it hurts also. Nice.
I'll wait for a second. So I would assume that the people online can hear me. So. Sure. No worries. Okay. All right. So everybody had a great morning. I'm going to try and do some filler. Maybe do a couple of notes confirmation with people online can hear. I don't have any prepared. Being a dad, I do have some. Like, so two men walk into a bar. You think one of them would have dunked. That's my dad joke.
I think one of them will what?
Would have done.
Right, takes a little bit of thinking.
That's pretty cool.
Anyone else have any jokes? No?
Do you mind just looking for that?
Well, yeah, if they do continuous manufacturing joke, that would be amazing. But, you have a joke? Go for it, Judy. Oh, that's a good one. And you see, I didn't get claps for my walking into the bar joke. So. Okay. Technology is really wonderful. So, when it works. I don't know. When does a joke become a dad joke? Good one. Good one. Excellent. Sure, you know what? Logistics are not something I think the people online will care that much about, so let me just go to logistics. So, just a notice to everybody, there is a recording going on, but it's for our wonderful, glorious note takers, so it won't be used for anything other than that, and will be deleted after 30 days. Hopefully it doesn't take us 30 days to write the notes. I just want to let people know that there is media on site, and feel free to talk to the media colleagues as you wish. And also, if you would like to engage with John Dobson, John can raise your hand. He can help you with any issues associated with the media. Something that we're going to be using today for collecting of questions, if you don't feel comfortable coming up to a mic and asking them in the room, is Slido, which will also be available for our online people. I'll have to go back and talk about that. We have two individuals who will be monitoring Slido throughout the day, and there'll be a way for you to anonymously, or put your name in, that's fine too, ask your questions through that system. Any questions that we don't get through, either from a presenter standpoint or the panel discussion, we will make available online with the question and the answer after the workshop. I would ask please to silence your cell phones, for while we're here in session. Restrooms, very important. If you go out either of the doors and follow the curve of this room to the left, you will find the restrooms. And from a safety standpoint, if there are any safety issues, a fire drill, etc., please follow the various exit signs, and USB staff will help direct you, out of the building. We're going to be using event timers for the presenters today. Green meaning you're good, yellow meaning you're coming up on time, and red for your overtime. And apparently we have 2 slides on Slido, just to remind everyone. We are using Slido today. I've gone through logistics. I've gone through jokes, and I've gone through logistics, so I'm going to wait for confirmation that I can continue. Anyone have any logistics questions I can answer while we're waiting that are not online? Is there Wi-Fi here? Yes, there is Wi-Fi. There should be a guest Wi-Fi. I will need to ask our coordinators to get the Wi-Fi password. I don't know it off the top of my head, but there should be a USB guest Wi-Fi access. And if not, I'll have them work on that for you. Oh, you can? Okay. Thank you, Tim. Tim does a lot more of these things than I do. So. Sure. So I heard some people had some travel delays. Hopefully everybody made it here with and are quite refreshed. I need to like write down for my lessons learned about have filler things ready. So in case something like this happens.
Hey Sam, do you know where this is at?
Maryland. I think it's at USB sets.
Okay.
Oh, sure.
Maybe next year you can get Anderson to pay for people to go.
Oh, they can get rid of the WebEx now? Oh, okay. Well, but that's a great question, Lawrence. Why don't I just start with that and I'll go into my slides fairly quickly. USP's journey within continuous manufacturing has actually been a fairly long, even though we've had a lot more effort in the last 2 to 3 years. You can actually trace this back to the idea of process analytical technologies, spectroscopy, and the new various techniques for monitoring quality that USB first started developing probably about 25 years ago, and developed some specific general chapters around that about 15 years ago, 15 to 18, something of that nature. And more recently, as USB started looking to the future and realizing that to advance the cause of quality and quality medicines, USB had to change along with the industry, and in some cases help lead the industry through that change. So advanced manufacturing technologies was identified as an area for USB to focus, to provide solutions and support for companies that wanted to use some form of advanced manufacturing technology. And through that process, continuous manufacturing was identified as the first primary technology that we would develop programs, best practice guidelines, standards, et cetera, around. And that all started with my good friend and colleague, Atul Dubey, who came in as our first expert in continuous manufacturing, and has blossomed from that particular point. So, good question, Lawrence, so thank you. From a continuous manufacturing perspective, and I just want to set this up for today, we've heard a lot about the various benefits that continuous manufacturing can have, either on a product or a company or an industry. From the standpoint of quality, environmental impact in terms of reduced waste. One of the areas that USP is rallying around, not just in the area of continuous manufacturing, is the potential impact that this technology can have on supply chain resilience. This became quite evident during the COVID pandemic with regard to the various challenges that supply chain has come out during that time. But the idea of a footprint of manufacturing that is smaller, more flexible, able to be distributed in multiple different locations is one that can help build that resilience of the supply chain. And there's one aspect of supply chain resilience that that as I reflect on today's agenda, how we developed it, that I think we'll take as a lesson learned that we've kind of missed a bit, is the idea that supply chain resilience ultimately is for the benefit of a patient and patients more broadly around the globe or in the United States. And in particular, just recently, in the last 9 months, there have been many news articles about supply chain shortages in some medications that are a bit surprising, especially in the area of cancer medications, but also in addiction, anxiety, pain medications, ADHD, have all had news articles around supply chain shortages. And for those who have worked with me at USP, for a while, know that one of the things I try to always instill is a sense of purpose, both into our employees as well as our stakeholders and partners that we're working with. And this is the area I want to try to anchor some of your thinking early on as we go through these presentations. Continuous manufacturing is a fantastic technology, but I would encourage you to think about, for you, why is this important to advance, from your perspective? And this is a personal thing. You can think about it, and it's for you. But why is the advancement of continuous manufacturing important? And just to give you an example, for me, here is a picture of my lovely wife, who is obviously thrilled that I told her I was going to be doing this today. You can tell that from her picture. But my wife's name's Kelly, and she has hypothyroidism. And she takes levothyroxine on a daily basis, and has for the last 40 years. And it's a chronic condition. However, if left untreated, it can go into some pretty bad areas, including coma and eventually death. For her, her thyroid barely functions on its own at this point, so she's dependent upon this medication. And so for me, making sure that we have a systemically resilient supply chain is very important. And I would mention that levothyroxine has been on the shortage list at least once every year since 2018. And so as I think about why I'm working at USP and I'm helping advance advanced manufacturing technologies, as well as in particular continuous manufacturing, this is where I come back to as to why this is important for me. And so I'd challenge everyone to think about that for themselves. So, and with that, we're going to go into the various keynote addresses. First, I'd like to welcome up Dr. Adam Fisher to the stage. Adam works at the Office of Pharmaceutical Quality at CDER and focuses on engaging FDA and stakeholders in supporting advanced manufacturing technologies. So, Adam, welcome up to the stage, and thank you very much.
Thank you. Thank you so much for having me. So you're probably wondering, why does this guy have 2 textbooks with him? And by the way, if these bright yellow textbooks are striking fear into your heart right now, then I know you're a chemical engineer. For those of you who aren't, I want to point out that about 25 years ago, I learned about the elements of chemical reaction engineering. On page 18, we have continuous reactor design, continuous stir tank, continuous plug flow. Bioprocess engineering. A little further into the book here, page 262, we have the design of perfusion systems and perfusion bioreactors. So this is not new stuff. Scientifically, not new at all. By chance, yesterday when I pulled this book off of my bookshelf for probably the first time in at least a decade, paper fell out. I had no idea what it was. I opened it up. This is actually my senior design project from undergraduate class. to design a continuous reaction for phthalic anhydride production, to design, financially justify, and support that reaction. So this is something that's taught to every chemical engineer, and certainly in this country and probably around the world. It's not entirely new. And I will point out that I see Jim Polly over there. So Bill Bentley was my instructor for bioprocess engineering at the University of Maryland. And then actually when I did my PhD at Cornell, Mike Schuler, who wrote the book, was my professor, also on my thesis committee. So I'm very happy to be here to talk about continuous manufacturing. I'm an advocate of advanced manufacturing in general. And I think to answer the great question that we kicked off with, why? For me, it's not about the technology. Right? I don't care all that much about continuous manufacturing itself in a vacuum. I care about the good that it can do. And I'm in the Office of Pharmaceutical Quality in CDER, that's the Center for Drugs at FDA. And this is what it's all about for me. Everyone deserves confidence in their next dose of medicine. Pharmaceutical quality assures the availability, safety, and efficacy of every dose. And I believe that advanced manufacturing technologies, in many cases, can do a better job. And that's why it's important to me. It's all about the patient and the impact on the patient. So let's talk a little bit about continuous manufacturing in general. So if you look at continuous manufacturing as a concept, it's generally most appropriate for processes with high repeatability. And I think pharmaceuticals are certainly within that. Right? High repeatability requirements, high control requirements for pharmaceuticals. And it may not be fit for every pharmaceutical manufacturing process, but it can bring some advantages in some cases. You heard some already. I imagine most, if not all, speakers today will have some version of this statement, but you can have better efficiency, reduce costs, smaller footprint, improve process control. And if you look, and this chart I have here is up through 2020, even though there aren't really that many pharmaceutical products made with continuous manufacturing, still they are growing, and a fairly substantial revenue is drawn from over $3 billion by the end of 2020. So not as niche, I think, as some people might believe it to be. Sometimes when people from other fields look at continuous manufacturing of drugs, they can bring their assumptions from other industries. So commodity chemicals, most that I'm aware of at least, commodity chemicals are made with continuous manufacturing. And the difference between pharmaceuticals and a lot of commodity chemicals, fine chemicals, and so on, is that the volumes and the runtimes can be different. So in the commodity chemical world, it is sometimes possible and sometimes financially feasible to have a continuous process that runs nearly 24/7, certainly over very long run times. In drugs, that's not often the case. I can actually think of very few, if any, drug materials that might truly justify a 24/7 operation, 365 days a year or something like that. It's just not the case. So the advantage for drugs is not that you can run it for a long time, but that the volume or runtime is flexible to meet a variable demand. Demand goes up, you can have longer runtimes, produce more volume. Demand goes down, you can have shorter runtimes, produce less volume. And as compared to commodity chemicals, again, which have very long runtimes, some pharmaceutical continuous manufacturing has actually been designed to reduce the runtime that you might have for a batch process. So it's a little bit of a different ballgame. You have this high value, low volume proposition in the drug world that you might not have in other chemical manufacturing industries. And I think one thing certainly that was driving the adoption of continuous manufacturing in the pharma space is that process equipment has become more amenable to the scale of drug manufacturing. So it's not just this super high volume, super long runtime thing anymore. There's process equipment that's available at more reasonable scales for pharmaceutical manufacturing. And there are some examples of this. You'll see a lot of references to continuous manufacturing in the scientific literature throughout my talk here today. And this is one recently published about a coding process for commercial pharmaceutical manufacturing in a continuous manner. I'm sure there's plenty others out there. So let's take a look at some of the benefits that we've seen, and certainly there are many, but I'll focus on just a couple here. So the continuous manufacturing benefits to companies. So one pharmaceutical company switched to continuous manufacturing and reported a 50% reduction in operating costs, 33% reduction in waste, 80% reduction in manufacturing and testing cycle time, and a 66% reduction in time from testing to release. Okay, and I think this fits in some of the statements that I just made about the difference between continuous manufacturing in the pharma space versus other industries. And so this is an example of a drug product, and another example related to drug substance considers the human element. Okay, so one company adopted continuous manufacturing for commercial Grignard reaction, which if you took organic chemistry and remember back to organic chemistry days, has a spontaneous heat release associated with it. So this can create a safety risk. And so this company reported designing a continuous Grignard reaction to be safe and well controlled, to require very little operator involvement to even improve that safety, and then to reduce the material movement between unit operations that involve the human in some way. And so I think these are two different examples of how companies in different spaces within pharma have seen benefits from continuous manufacturing. So we've already heard a little bit about advanced manufacturing, and what we're talking about here today is continuous manufacturing. I think it's important for me to emphasize that when it comes to FDA, advanced manufacturing includes more than just continuous manufacturing. I think certainly it's fair to say that a lot of our experience thus far has been in working with companies developing continuous processes, but there are many other types of advanced manufacturing technologies as well. Advanced PAT, multi-attribute method, novel container closure systems, and so on. So it's not just continuous manufacturing, but there's all kinds of benefits that come from adopting advanced manufacturing technologies. Better quality medicine. It's been a drive for a long time to try to realize Six Sigma in pharma. And I think other than some rare instances, it's probably not the case. So I think there's still a drive to do that. I think advanced manufacturing can help. We heard earlier about reshoring drug manufacturing facilities. Certainly that's been a very hot topic over the last few years. Developing drugs fast, so speeding the development of novel or patient-focused therapies. And then certainly preventing drug shortages. We heard about some shortages earlier related to cancer drugs that are out there, and there are unfortunately many more examples that we could point to that have really impacted patients in a significant way. One of my, I was going to say favorite, but perhaps it's actually my least favorite statistic. We did a survey of healthcare providers, and over 80% of cardiologists and dermatologists reported that their patients had had trouble filling a prescription due to a drug shortage. This was pre-COVID. Okay, it's pre-COVID. 100% of pharmacies reported issues related to filling prescriptions due to drug shortage. This is not an acceptable scenario. Right? And so then, obviously, last on here, and I'd like to point out, I use this slide even pre-COVID, but improving our emergency preparedness. Right? I mentioned how continuous manufacturing can help in flexible production. So if you have a big spike in demand, you could potentially scale up your process very quickly if it's a continuous form. And I'll talk a little bit more about that later. So I'll talk a little bit about what we have done, what we do in Cedar to facilitate continuous manufacturing adoption. So the first thing I want to point out is that we're not the only game in town. Okay, I am here representing Cedar Center for Drugs, but the Center for Biologics, CBER, also has a very similar program to the one that I'm about to describe here. And certainly advanced manufacturing as a concept, as an initiative, has been very important to the agency overall. But I'm involved in CEDAR's Emerging Technology Program, and I want to talk a little bit more about that with you here today. So 2014 to me was a very important year to continuous manufacturing. I see that as a year that we first saw a critical mass to really drive the adoption of continuous manufacturing in pharma. And I think it's no coincidence that right around that time, Cedar also established the Emerging Technology Program to facilitate emerging technologies like CM. And what makes the Emerging Technology Program very unique is that you have representation from all of the quality assessment and inspection program at the FDA. So you're not just talking with a niche group of folks who then, once you submit an application, you're going to pull in many more people that have no idea what's going on. You have representation from all of the relevant programs from the beginning. The other feature that makes it unique is that you can have early engagement before and during submission and assessment. Okay, so it doesn't have to be once you submit an application, then we can have some conversations. Actually, it works better for everyone if we do it before you submit an application. That way, we're not caught off guard, and we can do our best to communicate with you about how to best prepare a submission. And this is really important. I want to emphasize this, because some people have gotten this wrong too. But these early interactions don't even have to be specific to a product or a regulatory submission. So you don't have to come in and say, I have this continuous process to make this specific drug. It can be more general than that. We can have conversations prior to identifying the product. And certainly, as I mentioned already, prior to a regulatory submission, it's better if we talk beforehand. And there's a guidance out here. Others may disagree with me on this, but I think it's fairly straightforward to apply to being in the program. It's a relatively simple 5-page application where you answer very general questions like, why is your technology novel? How will it help patients and quality, stuff like that. So hopefully it's not too burdensome to be a part of that program. And so because of this program, I think we've been able to have some successes in continuous manufacturing. So I mentioned it started in 2014. A year later, 2015, we approved the first continuous manufacturing submission for a small molecule drug product. A year later, 2016, we approved the first switch for a batch product to a continuous one. And this was for a drug product. And we're now at the point where we have accepted over 50 continuous manufacturing proposals meaning to be in the Emerging Technology Program. And we've approved a diversity of submissions, 20 total now. And when it comes to CM, I just want to point out, even though there may be still a relatively small number, they do cover a fair breadth, right? So we have a drug product employing CM for API synthesis, okay, so drug substance. We have a continuous biomanufacturing process, so not just for small molecule stuff, for biological products as well. I mentioned already a switch, but there's a drug product that can be made using either a batch process or a continuous process. And then we also have a case where there are multiple drug products, 3, that can be made on the same CM line. And we just want to pause and emphasize that for a second, because a lot of people have come to me and said, well, continuous manufacturing, you have to design it for a product, and it's not amenable to having other products made in that line. And it's not true. I think it's already been shown that you can produce multiple drug products on the same CM line. I mentioned 2014 was a tipping point, and I think one of the reasons that 2014 was the tipping point was because they had the first, and I imagine some of you even went to this, the first international symposium on continuous manufacturing pharmaceuticals, to bring a number of different stakeholder groups together to discuss the opportunities, technologies, and regulation of CM. And the 4th symposium was just held in April 2023, and a big focus of that was celebrating the accomplishments in continuous pharmaceutical manufacturing over the last decade or so. And one of the really neat parts of the 2014 symposium was that they put out a series of white papers on different topics that they felt we needed to collectively address in order to push the adoption of continuous manufacturing in pharma. And they're all here, there are a lot of them, but I will just highlight, to me, some of the important regulatory concerns that came from this. So one was definition of batch. And for a continuous process, how do you define a batch? A lot of the regulations are based on batch and so on. Process dynamics, how do you measure and control? Changes in production output, how do you handle that? And then continuous process verification. And so what I think is neat now, if we look back, we just recently released the internationally harmonized Q13 guidance on continuous manufacturing of drug substances and drug products. And it really almost systemically addresses some of these issues that they outlined back in 2014. Right? So what's the definition of a batch? We can define it by the quantity, output or input material, or by runtime. How do we handle process dynamics? Well, we have sampling diversion strategies. Some examples of what that might look like. How do you how do you handle changes in production output? Well, you can look at handling that by runtime, by flow rate, by scale up, by scale out. There's ways that you can handle these things. And then also there's a dedicated section on continuous process verification. And I also want to point out here that US regulators aren't the only people in this game. Other international regulators have already approved the SCAM applications. So Canada, Europe, Australia, Japan, Switzerland, New Zealand, I'm sure there are some others, right? But these are some of the ones that have certainly been more public with what they've done here. So I think we're making progress, internationally harmonized guidance, and we're doing a lot of things that address the issues that were originally identified back in 2014. So mission accomplished. Why are we all here today? Well, I think there's more. I think there's more that people are doing, and certainly more adoption that we can facilitate. And one thing that I think is coming next is this concept of end-to-end continuous manufacturing. So I gave you my examples earlier of things that we've improved. We have experience improving continuous drug product. We have experience improving continuous drug substance. But end-to-end continuous manufacturing is the marriage of the two with no stop in between. So straight through drug substance to drug product with not necessarily an isolated intermediate there. And I think these end-to-end processes can bring different regulatory considerations and maybe even challenge the regulatory expectations of the existing framework. Then I think the other element that's going to be coming soon is marrying continuous manufacturing technologies with other advanced manufacturing technologies, right? And a lot of what I'm pulling here are quotes from this National Academies study that we sponsored on innovations in pharmaceutical manufacturing on the horizon. And this looked at a 10-year window and told the FDA what we could expect to see over the next 10 years. And so one of the things that they pointed out was continuous manufacturing units that use artificial intelligence to enable adaptive process and product quality controls. And so I think there are even other examples of technologies that might work together with continuous manufacturing in the future. And I'll talk about some of those in just a second. But I think this is our challenge now. We need to prepare for the future. We've dealt with things through the present, and now we need to prepare for what might be coming next. And so one of the reasons I'm here today is not because I am the foremost expert in continuous manufacturing. It's actually because I am the lead of CEDAR's Framework for Regulatory Advanced Manufacturing Evaluation Initiative. And what we try to do in Frame is to identify risks related to the regulatory framework and address them such that innovative and promising advanced manufacturing technologies aren't stymied by a regulatory framework that's not compatible with them. And advanced manufacturing can be anything. Well, maybe almost anything. And it's really difficult because we can't consider everything or we won't get anything done. So we focused on 4 priority technologies. And a lot of these technologies came from the NASA report that I mentioned already. And so distributed manufacturing is one, and this is where we have decentralized manufacturing, where it's not at one large-scale traditional brick-and-mortar location, but manufacturing is distributed in more geographically diverse and smaller-scale locations under the control of a central site. Related to that, point-of-care manufacturing. This is manufacturing close to the patient, maybe at a hospital or within a healthcare facility, something along those lines. We have end-to-end continuous manufacturing, which I mentioned already. I do want to emphasize that even though this is a new thing, it was addressed somewhat in Q13 already. So Q13 devoted an annex to the unique scientific and regulatory considerations of an end-to-end process for solid oral drug products. So already some regulatory framework shaping in this space. And then I already mentioned artificial intelligence. This is something that's really growing. If we said 2014 was when there was a critical mass for continuous manufacturing, I think 2020, in 10 years, 2023 might be considered the critical mass for artificial intelligence. Not only in pharma, much more broadly than that. So these are our 4 technologies, and we're doing a lot in this space. Some of you may have already been involved in this. Some of you may be hearing it for the first time. But what we've done for distributed manufacturing, point of care, and then also separately for artificial intelligence, we put out discussion papers on these. So in these discussion papers, we laid out some of the issues related to the adoption of these new technologies as we saw them. And it's a discussion paper because we asked for comments from the public, from stakeholders, to help us know, have we identified the proper issues? Have we identified all the issues? Is there anything else that we think we need to look at? And so there was an opportunity to do that in response to Federal Register, where the paper was posted. But then we also held a distributed and point-of-care manufacturing public workshop last fall with PQRI, the Product Quality Research Institute. And this year we're doing the same thing with artificial intelligence in manufacturing. So we released a discussion paper earlier in the year, we asked for public comments, and then end of September, we're going to be having another PQRI workshop on artificial intelligence in pharma manufacturing. And we take this opportunity to invite any of you who are interested in artificial intelligence in pharma manufacturing to attend that workshop, join in those discussions with us. I already pulled out my old textbooks, okay? So hopefully it emphasizes the importance of science as the basis for what we're doing. And we can't generate the knowledge that we need to handle advanced manufacturing technologies without science and research that supports the knowledge necessary to drive it forward. And we have a large number of intramural and extramural research projects related to advanced manufacturing. Some of these relate to continuous manufacturing, not only in the small molecule space, also in the biotechnology product space. We see a continuous perfusion bioreactor. This is at our FDA laboratory right down the road in Silver Spring. Continuous perfusion bioreactor, lab scale. I have a couple other continuous bioreactors that we use for research and modeling and so on. And then some of our extramural grants as well focus on artificial intelligence, process analytical technologies, modeling and simulation, training, and so on. And so this is a very important space for CEDAR and the research fund. And I think really to emphasize that, you can go and look at, well, I can go and look at the full number of research projects within CEDAR. And so advanced manufacturing is employed for certainly less than 1% of human drugs, but greater than 10% of CEDAR's overall research projects address advanced manufacturing. And so you can see that here, we have about 28 projects that focus on continuous manufacturing specifically. We have some that focus on artificial intelligence and machine learning, and then also a handful that focus on 3D printing. So it's a very important research space for us. This is what enables us to have effective engagements in the emerging technology program and come out here and do some of the things that we're doing here today. But these aren't just sort of fun research things where we get to publish a paper and everybody pats us on the back, right? The idea is really to drive regulatory actions. So this research helps us form regulatory recommendations for the industry to guide their product and technology development, for example, through the Emerging Technology Program. It informs new standards, guidances, and policies. The Q13 international guidance is a good example of that, and also materials for training our own staff, right? Because you may have a relatively focused group that tackles a new technology, but then eventually you need to pull more people in to be able to handle that as the technology is more broadly adopted. I'll talk a little bit more about that later. I do want to give some specific examples though, because these are some general statements about what we can do. Emerging Technology Program, right, fueled by some of these things. Directly informing the assessment of an application. So this is the person who pulls up the application and then makes determinations on improvability. Some of that is based on their knowledge that they get through CEDAR research. I mentioned Q13, but another one that I want to hit on is the Q5A guideline. This is related to viral safety evaluation of biotechnology products, and they're tackling the continuous manufacturing aspect of that at the moment as well. And then a big internal effort that we have relates to process models. And using process models that can aid in the assessment of CM submissions. And I just want to tout that some of our researchers were awarded the AICHE award. That's the American Institute for Chemical Engineers. They're awarded for excellence in integrated quality by design practice for their use of modeling for the quality of risk management for CM processes. Some of these things have been incredibly helpful in engaging with companies who are developing these types of technologies. So my hats off to our researchers for doing these types of things. All right, let me talk a little bit about moving the regulatory hurdles. As the lead of CEDAR's FRAME initiative, this is what I'm all about, trying to remove the regulatory hurdles so that we can see our why be addressed and facilitate the adoption of continuous manufacturing on a broader scale. So I want to start by just dispelling some myths with CM, because people come up to me and get this wrong often. So some people have said, well, FDA is pushing for continuous manufacturing. They're trying to make us manufacture in a continuous manner. Let me be really clear, that's not what we're doing. Okay, the FDA will approve drugs if they comply with the appropriate standards, regulations, and laws. That's what we do. It doesn't matter how they're manufactured. Other people have said, well, FDA thinks everything should be made with advanced manufacturing, continuous manufacturing. So let me be real clear on that. That's not what we're saying. Okay, we think that the benefits of AM and CM can be realized in some, but not all instances. We don't believe that everything should be made with advanced technology. Another myth that we hear is that, well, CM is just for innovator companies. It's not for generic companies, maybe not for biotech companies. Let me be clear, that's not true. Right? Continuous manufacturing can be for, and advanced manufacturing certainly can be for generic companies, biotechnology companies, and so on. And we have had engagements with generic drug companies in our emerging technology program already. Okay, so it has happened. Another concern we hear from generic companies is that if innovator companies adopt advanced technologies like continuous manufacturing, then they will be required to use continuous manufacturing. Let me be clear, that is not what we're saying, right? The specification criteria are based on patient relevance and not on process capability. So just because an innovator company has used a continuous process, it does not mean that a generic company also needs to use a continuous process. And then finally, one I really want to highlight here is that we have heard over the years that companies are afraid to adopt continuous manufacturing because it will take the FDA longer to review and approve their applications. So we did a look. We said, okay, is this true or not? We have enough where we can actually pull some numbers and determine if this has been the case. So we published a paper on our self-audit of our continuous manufacturing applications that we received. And I was even surprised at this, okay? The CM applicants had shorter times to approval and marketing as compared to batch applicants. So if you look by median time to approval, CM applicants were 3 months faster to approve. If you look at the median time to market, CM applicants were 4 months faster to market. This is from the time of submission. And months translate into dollars, of course. So if you look at the revenue, the monthly revenue for some of these products, this is translated into not just a little bit, but hundreds of millions of dollars of monthly revenue for these companies by this shorter time to approval and marketing. I want to focus on time to marketing though, because I think that's an important one, because they already mentioned that one of the benefits of continuous manufacturing can be easier scale up or scale out. And when we looked at the time between approval and marketing based on median, the number was 0 months. Okay, so essentially, as soon as the product was approved with a continuous process, they were ready to scale up and hit the road and get going. And so I think that's a very important thing not to overlook, the time to scale up. And then we looked at some other things, because we hear a lot of concerns related to, well, it's going to be really hard for me to change anything in my continuous process once it's been established. Or pre-approval inspections will be a real hassle if I have a continuous process. And we looked at some of the elements related to these two factors, and we didn't see anything incredibly concerning or certainly substantial between CM applicants and batch applicants. So hopefully this goes toward dispelling the myth that it takes longer to approve CM products and it's more difficult to approve CM products. And actually, it has been the opposite. We have approved them faster and they've hit the market faster. Small sample size, of course, but I think it's a good sign for things to come. And one of the extramural research projects that we funded was to look at the costs associated with investing in continuous manufacturing. We funded a financial risk expert from the University of Maryland Business School. I understand we have some University of Maryland Business School people here today. Looking forward to hearing you talk later. And it looked at the costs associated with brand and generic companies investing in CM. And this was in foreign or domestic manufacturing. And what he reported was that continuous manufacturing pharmaceuticals is more economically attractive in the USA for a new facility than foreign manufacturing. So if you invest in CM in the US under current tax rates, there was a positive net value over batch facility in China or India. And this is for both brand and generic companies. Okay? So I think this was an important finding, and I think it's got a lot of airtime. So this was Professor Cliff Rossi. I know he's been out to speak to a number of pharmaceutical groups about this, and it's certainly something that we'd like people to be aware of as well. So when it comes to a regulatory framework, okay, I want to show how I think it can be done well for a new technology by using continuous manufacturing as an example. I talked through some of these already, but I just want to emphasize the timeline here. We heard that manufacturers were hesitant to adopt CM without additional engagement from the FDA. So we created the Emerging Technology Program in 2014 to allow for additional engagement. We heard that manufacturers were hesitant to adopt CM before the FDA had shown they could approve a product manufactured with CM. That happened in 2015. As I mentioned, 50 proposals from the industry since then, and 14 additional submissions since then. Then we heard manufacturers were hesitant to adopt because they couldn't switch over a current batch process to a continuous one. I already mentioned that happened in 2016. We heard manufacturers could adopt CM without guidance from FDA. So we released draft guidance on continuous manufacturing in 2019, served largely as the basis for starting international harmonization. And also want to emphasize the Q5A guidance on viral safety evaluation of biotechnology products as well. That's been another important part of this. Then manufacturers were hesitant to adopt, as I mentioned, because they thought it would take a long time. They feared that the timelines would be too long, and we showed that we actually approved the CM applications faster. And then finally, we said this is all great, but we can't do it without this internationally harmonized guidance. And just this year, we were able to release the internationally harmonized guidance in 2015, which, as I mentioned, also includes an annex on end-to-end continuous manufacturing. So I think this is an example of how you can adjust the regulatory framework in a way that removes the hesitancy of manufacturers to adopt a new technology like this. And so part of this was discussed just last month at the Duke Margolis Center for Health Policy. We had a workshop on innovative manufacturing approaches. And I just want to highlight some of what we heard there. And this is what one of the participants said in the panel discussion. Everybody said more or less the same thing. The regulators will never let us do it. We went to the regulators. FDA loved the idea. I believe the FDA has done a phenomenal job of promoting adoption of advanced manufacturing, supporting industries, and leading other agencies. And obviously, I highlight this point because it makes us look good, but I think it also emphasizes that there are other things that need to happen to facilitate the adoption. You know, some examples they gave were making a stronger economic argument, right? Doing a better job of selling to the C-level companies, getting more people on board to support the adoption of overall in the industry. So I think it was overall really nice workshop to be a part of, and I was happy to be on this discussion. Let's talk a little bit about the future, right? So what does happen next? An emerging technology cannot be emerging forever, right? So how it's supposed to work is the industry develops an emerging technology, it's accepted into the emerging technology program, we evaluate the technology, we have ongoing engagement with industry until they have a submission, and then after there are, this is not a hard and fast rule, but after there are 3 applications that have been received from 3 unique companies, it can trigger a graduation event where the technology moves from the emerging technology program to the standard quality assessment process. And this is an important part of the life cycle of new technology. It can't rely on a special program to handle it forever. At some point it needs to be handled by everyone, the agency. And so this is important, and certainly training and knowledge is an important part of making that possible. So we've already begun to implement the next iteration of the Emerging Technology Program to improve our strategy, to improve the operation of the program, and then also to improve the communications with developers of new technologies. And eventually, all technologies graduate from the Emerging Technology Program. And I'll give you an example of one that has already graduated, which is continuous direct compression. I heard continuous direct compression described once as an inherently continuous process. As long as you are feeding powder in and it's being compressed, you can kind of keep that going for as long as you need. And so continuous direct compression has been something that we have now dealt with enough, and we have done enough training of our reviewers that it's not necessarily eligible for the Emerging Technology Program anymore. It can be handled by any of the staff, any of the quality assessment staff in OBQ. And then another part of the future is new legislation. New legislation bringing new opportunities. So we have PDUFA 7, that's the Prescription Drug User Fee Act, and then also what some call FDORA, this is the Food and Drug Omnibus Reform Act. And these aim to advance manufacturing technologies as well, asking the FDA to issue guidance, to hold a public meeting on the designation of advanced manufacturing technologies, which is actually what we did at that Duke Bolus event that I just mentioned, and to establish the Emerging Technology Program in statute. And I think there are maybe even some other things that this type of technology brings. One baked in here, there's an Advanced Manufacturing Technologies Designated Program, and then there is also a Platform Technology Designation Program. And I think those 2 programs will need to be designed very carefully to encourage people who are developing innovative technologies, but not be so broad as to lack value in media. And that it's going to be a challenge to do that, but it's something that we're tackling right now. So, just want to close by saying, we will continue working with stakeholders. We want to facilitate the adoption of advanced manufacturing technologies, such as continuous manufacturing. And our why is because we want to better assure the availability of quality medicines to patients. Thank you. I'm almost out of time. Should I do? Yeah, I can maybe do like 1 or 2 questions, something like that. Yes, sir.
Question about the graduation. It's clear that the FDA is graduated from the systems of vision, job, results. What's it really it's a small data, rules, and everything that don't have flows that love to have mechanisms to do conversations and eventually would there be other mechanisms in place to support that?
Yeah, so a couple things I want to address there. Long history of continuous direct compression research and support in academia as well. So I think there's a rich source of knowledge that companies can tap into outside of their own walls related to continuous direct compression. And the second thing I do want to emphasize, when I said it graduated, I believe I used generally a couple times. There are still cases where continuous direct compression would be eligible to be in the Emerging Technology Program. It's on our website. if you go to the Emerging Technology Program website for FDA, you can see there's a table that lists things that need to happen in order for a CDC technology to still be in the Emerging Technology Program. So there is still space for people to be there. Yeah. All right. Thank you so much.
Well, thank you so much. Next, I will like to invite Dr. Todd Maloney from Eli Lilly Corporation up. Todd is the Executive Director in the Synthetic Molecule Design and Development Group at Eli Lilly, and really looking forward to your presentation today. And they tell me that all you need to do is open it up and I'll switch it over directly to you. So, good. So, thank you, Todd.
Thank you, everyone, for the opportunity to be here today, especially my company, Eli Lilly. Today I'm going to share with you a little bit of the history of PAT, continuous manufacturing, kind of an endeavor that we've been pursuing for some time at Lilly, as well as follow up with a case study, just kind of highlighting some examples of how we've applied some of this learning to a small molecule synthetic process that was developed a couple of years back. And if I have time, maybe just give you some metrics on a recent process. With that said, opinions that I share about PAT and continuous manufacturing today are my own. It may not reflect everything that we're doing within the walls of the Eli Lilly company in the space, as we have been active for quite some time, as this first slide shows. So, as early as the 1980s, process analytical technology, if we want to call it that, was actually being utilized in commercial processes in Indianapolis and Puerto Rico. And the photograph that you see there, if you've been in an analytical lab a very long time, that's an HPLC with a strip chart report, okay, which doesn't predate me too much, but just a tad. But it really highlighted, that there are applications, even going back almost a century, or 50 years now, sorry, that could potentially benefit from having some type of analysis being performed, in-line or online. It rapidly accelerated in the mid-2000s and into 2012, 2013, when we saw 3 hybrid processes that are executed in our manufacturing sites in Ireland. This really catapulted our investment into this space, culminating in most recently in 2017, the launch of a small volume continuous facility in our manufacturing suite in Ireland, and then a rapid expansion of that facility within the last 2 years to take on some modalities that have gone through some hybrid continuous type of processes. This facility was truly an honor to be able to work and partner with our colleagues in manufacturing to launch this. Right? It was multiple years of development work, partnering with our scientists in manufacturing, both quality, engineering, QC, and then across all the process development. It really, truly was a team effort. And so to see it, being launched in the operation was a really remarkable thing. And that's kind of shown here, and this is also recognized by the FOIA from ISPE, receiving Affiliate Facility of the Year award in 2019. But just a year later, in 2020, facility that we built in Indianapolis, Indiana, which is the home of the Synthetic Molecule Design and Development Group, was also awarded a FOIA award. It is a novel facility. It kind of dates me to think that we've been in our building almost 5 years now, but it just seems like yesterday it's so new inside stuff. But that's really where the heart of innovation takes place within our company in the synthetic molecule space, with ultimately the goal of translating that knowledge, translating these new capabilities, this innovation into manufacturing so that it can truly and ultimately benefit patients. And so at the heart of this facility, right, for small volume continuous, is this concept, right, of linked unit operations, right? So being able to link synthetic steps together so that we can telescope, accelerate, and address a lot of the attributes that we heard Adam talk about in terms of what are the benefits of implementing continuous unit operations. Not every process that contains continuous unit operations is really continuous, right? There may be hybrid steps. We may decide that a particular reaction can benefit more from being operated in a batch process. And then ultimately, we may incorporate a variety of different analytical tests within this process, right? Some of it might be PAT, some might be at-line, some might be completely offline, right? Each process is going to be different and have its own attributes that need to be controlled based on chemistry as well as the unit operations that are ultimately implemented to make the particular material. So over the years, we've successfully had installation of a number of different PAT tools within our manufacturing suite. Some of these have been applied to batch processes, but a majority of them have also been applied to hybrid or other types of continuous unit operations. So, tools such as chromatography, mass spectrometry, FTIR, we've done some near IR, GC, all the way down to simple parametric sensors like refractive index, electrochemical, things that aren't necessarily monitoring something's specificity or selectivity per se, but they can definitely tell us when a change or some type of variance has occurred within that particular unit operation, and it may or may not require the attention of somebody to go visibly look at that. But what I'm going to focus on today in my case study and kind of share with you, is some of the work that we've continued to do in the space of online chromatography and why we believe it's been such a valuable asset in PAT tool for us as we develop both continuous as well as hybrid processes. And so, as I kind of mentioned just a second ago, right, we've kind of bucketed PAT tools into three different spaces, right? So, an outline PAT tool isn't necessarily a PAT tool per se, right? It's just an instrument which is proximal to the process where if a sample can be pulled by an operator and an analysis can be performed on a rapid basis and information can be generated that can then be used to make a decision related to that process. Online is something that we utilize extensively, right? And that's what I'm going to talk to you a lot about today, where we're physically removing the small aliquot material from that process. It's not going back, but it's going to be destroyed during the analysis so it can generate information that can then be translated to knowledge of how to get a step change potentially within our process. And then you have inline PA tools, right? So where you have a sensor or some other device which is completely immersed, within the process, it's not taking a sample per se, but it can provide very high frequency and high dynamic information back to the process, right? As processes are designed and developed, each one of these tools can provide different levels of control or potential control to your process based on how that knowledge is generated and applied to decision making. But what really put Lilly on the map and really gave us this opportunity to use a large instrument, right? LC is not small, right? It gets into all types of discussions on, do you really want to have this in your manufacturing? Or do you really want to have this in a field? Is this something that is amenable? Can't we develop a simple sensor? Can't we develop a model? Isn't there something else we can do that's easier than chromatography? And the answer is yes. But when you work with chemical engineers 24/7 who want detail, specificity, selectivity, they want to understand performance of every single one of those unit operations, you can start designing processes that are amenable to something like chromatography, which from a PAT historical perspective is a slow tool. It's not fast. A chromatogram that takes 2 or 3 or 5 or 20 minutes is slow compared to the dynamic response time of a Raman or near IR or inline UV. But the specificity and selectivity is king, and hopefully I can share with you some of that here shortly. One of the other key things that we've had to develop over time is this ability to manage the data. Right? That was always the big crux for us, is we were finding out that yes, we can put sampling systems online, we can connect high resolution with slower techniques like HPLC to our processes, but how do you get the data? Right? Because what we found we were doing is we were getting all this wonderful online data, and then we were manually crunching all of it, and then trying to figure out how to get it to the engineers and to our colleagues so that they can make decisions off of that. So not really intuitive, right? We're just moving the bottleneck, if you will, in the whole process. So we developed this really elegant process where we partnered with a couple of different software companies and instrument vendors, where we basically have the ability to send a command from our distributed control system, which in this case was Foxborough, Delta V, any of the other products that are out in the market in that space, where it tells the dilution cart that I showed on the previous slide to pull a sample. That sample then gets delivered to our online LC. That online LC runs an injection using an instrument method that's been installed and qualified. It generates chromatogram and data. Once that data is generated, this control system then initiates an orchestration in a tool called SyntQ, which is a PAT data management tool that we use in line. It initiates that analysis. That data is then auto-processed, exported, it's sent to our historian, and then once it's in that historian, it can be trended, dashboarded, historized with any of the other parametric or process control data so that we can see the analytical data in real time with the engineering data, right? From our perspective, that's something that's really important and powerful being able to really see the value of process analytics, whether it's in development or all the way up through, a true manufacturing process. So what I'd like to talk to you now about is a case study, kind of looking at a small molecule continuous manufacturing process that we developed a couple of years ago, where PAT was a crucial part of that whole process design and ultimately elevating that process, into our manufacturing suite. I will say that I did not coordinate some of the chemistry I'm going to talk about at all with something Adam showed on his slide, but the irony of it is pretty neat, and hopefully you can see some value here when it comes to linear reactions. So this is a continuous unit operation that was developed, for a small molecule asset in our portfolio. And as you can see here, this is a multi-step process where we have a number of different unit operations connected, a number of different platforms that we use, so both what we call PFRs or plug-for reactors, as well as CSDR, continuous stir tank reactors. And the slide is a little busy, but you can see where PAT has been implemented here across several different steps. But please also notice the offline analysis, right? Again, complementary ways to measure and integrate and use analytical data, right? We really are, designing processes and trying to understand where the best utility of these tools is, not force fitting it into every single unit operation because we can't, right? It's got to be used in a situation where it can ultimately help generate that process. And when we look at these continuous unit operations, when it comes to residence times, right? So not just necessarily in the chemical transformations that are occurring across these different reactions, but in the time that we're, basically sitting and holding material before we forward process it into that next step. That's captured here in these green buckets, right? As you can see from the slide, we have residence times on the orders of several hours, all the way up to almost a day, maybe even longer in some instances, right? These residence times are really helpful in how we design our PAT strategy and how we can measure and monitor some of the quality attributes of each of the steps throughout this process. So an example of that is shown here in this first slide. So this is the step 3, first step that I showed on the previous slide where you have this transformation occurring, starting material to product. And this is the online chromatography data. So this is a results table. Right? So if you're an analytical scientist, you're used to looking at a report and seeing a table of components identified and an area percents, or a quantitative value if you're doing quantitative analysis. But what we can rapidly trend here in our distributed control system is this peak area data, right? This relevant for a given unit operation, particular type of analysis. So we can see our green trace at the top of this trend is a product, for this particular conversion. We have the peak area of that product also plotted here, as well as our starting material and some of the impurities that we monitor across this process. So, given that this is the first time that we were able to deploy and run this in this manufacturing environment, this is pretty exciting to us as well as all of the people on the process team, being able to see that analytical data trended in a dashboard setting, alongside some of our other process data. And that's what this slide here shows now that we introduced the trending of our flow rates. So you can see here we have 2 pumps delivering starting materials and reagents into this process, and we can plot that data alongside that peak area data. So if you're on the plant floor and you're monitoring a unit operation, this is really powerful and exciting to see, right? Where we have analytical data mirroring the process data and showing trends that we observe in that. We can even look and monitor shutdowns, right, or intentional holds with that process. So for this particular transformation, we intentionally had several holds designed and implemented into this process to develop better process understanding for this unit operation. And we can see here that the analytical data obviously no longer exist because no new material is being generated and pushed to be sampled and then just transferred over to our online HPLC for analysis. The next unit operation or reaction in this particular sequence, right, is a Grignard reaction. And as we heard, there's a lot of interest in these types of reactions. They're very powerful for doing some novel transformations in small molecule synthetic chemistry, but obviously there's some concerns around potential operator safety and overall just being able to control and manage these at a larger scale becomes a little more challenging when we talk about doing it at a manufacturing scale. For this particular step, we had periodic charges of magnesium approximately every 12 hours, right? So again, understanding the exotherm and being able to monitor this process is important for a lot of different both safety and potential quality attribute implications. the quench vineyard product is analyzed by HPLC. And one of the really key elements of this, right, is that we had to do a lot of development studies, right? This wasn't just something that we switched on and said, hey, we're going to run it, let's see if it's good, right? An exceptional number of experiments were run to confirm, one, that we had successful conversion during this process to the proteome, without ensuring that we were precipitating solids, right? And the reason that's important is because those solids, right, those fines that could be generated in this process, if they are drawn into our dilution sampling system, could potentially foul some of the components in that, or if they make it through that sampling system and are delivered to the online LC, potentially foul the plug tubing there, which would prohibit us from sampling. So we did a lot of work to understand this and be able to develop and control that. And so if we look at this particular step, look at our online HPLC data output, right? We can again see a product trend, we can track a well-rejected impurity that we know can exist in this process to make sure that it's staying at a level that's appropriately rejected downstream. We can also see one of our starting materials here, right, where you see this trend, right, where you see it trending upward, and then we have a magnesium charge initiated, you see it come down, you see it come up, you see it come down. Right? This is classic vineyard chemistry, right, where we're charging for magnesium consumption in that starting material. So this is, again, just a really novel and exciting thing to see as an analytical science and working on that floor. But as we heard previously this morning, something else that's very important to understand, though, is even though we're seeing depletion and then addition of this magnesium charge to this process, the exotherm stays relatively flat throughout that process, right? So even though we're depleting, and then adding back more of this magnesium to trigger this chemistry for the starting material, we're still not generating any type of situation where we have safety concern for any of our scientists or operators working in the plant. So very important attribute of PAT beyond just measuring product quality. Another aspect of this particular unit operation, is that when it starts up cold, right, we have this whole time, of the CSTR, reduced temperature at the beginning of that process, right? We can start sampling chromographically throughout this process, right? And we know what the targets are for the analysis. We know what the quality of the time to derive out of this. So we can generate that online LC data and kind of watch as this process start up. And because we know that this material will not meet the downstream purity profile that's required for us to fully process it, we can just divert that material completely out of the train until we start seeing purity of that material that's acceptable. We can then divert that process back into collection and now collect over that extended period of time. In this example, these are days and weeks. Right? This is not like a 24-hour time cycle. This is a long-running process where we have a long residence time for some of these unit operations, as I showed on that earlier slide. But one of the things that's really important to understand with any of these unit operations, right, is the impact of base impacts, right, disturbance. As I talked about, there's two predominant platforms that we use in this protein process to generate some of these applications. One is a plug flow reactor, right, which is as simple as it looks. In this example here, showing the photograph, it's a PTFE2 coiled in a tank that we can control with liquid to maintain a constant temperature for a given unit operation. And when you flow liquids through an open tube, right, if you've ever done open tubular chromatography, pretty straightforward. It flies and comes out fast, right? And you get this really nice narrow residence time distribution. You get this pulse that has a very Gaussian trend because of the plug flow profile that you have within that open tubular system. When you look at a continuous stirred tank reactor, right, which you could argue is kind of like a mini batch reactor in essence, but with the exception of that we're feeding in materials and feeding out product. Here you have a much broader residence time distribution, right? And what this can do, if you look at the overall trend of material transfer in and out of the system, it kind of generates this pulse, right? Where you have this tail, and inherently this can potentially lead to more carryover, if you will, in that system, right? And it generates what we call this residence time distribution probability, where we're trying to understand the amount of time the material spends in that process, and then what is the ultimate impact of how we sample and potentially control the disturbance within that process. So to do that, we have to understand divert, right? In our process, you saw that we have surge that's in several of these unit operations. The surge provides us obviously the opportunity to generate material that can be forward processed to the next unit operation so that it can run successfully, but it also gives us a really good opportunity to have time to analyze material, either online in terms of PET or offline. Sorry. that caught my eye. I went off the screen. But also to be able to, generate data to investigate a disturbance that may occur in the process, and then understand what the downstream impact of that disturbance is. So we came up with a flow chart here, based off of, some of the guidance and literature, where when we detect a disturbance in our process, right, can the disturbance be mitigated, right, by the process? If the answer is yes, then we collect the downstream surge, move forward and carry on with our process. If the answer is no, right, we inherently have two options in this particular example. One is to divert at the source, right? The other is to divert at a downstream surge point. For either of these scenarios, right, the process risk assessment has to be performed to determine so you and install the appropriate strategy to mitigate that risk. PAT is also extremely powerful and enables us to rapidly detect that and help us decide what the effective management of disturbances in that process. So even a slow 20-minute analytical method can help us do that for those particular types of human operations where we have these longer residence times and these surge points. So for this particular example, the divert limits were based on rejection studies to determine specs or impurities of concern at the API surge solution, right? So we had drums that were being filled at the end of this unit operation about every 10 to 14 hours. And we could then go sample those drums offline or offline in addition to KP data that was being generated online. Divert limits, in this particular example, we're defining as a level of particular impurity that could raise failure of an API surge, right? And if it persisted for the entire surge collection time. So not necessarily an instantaneous disruption, but a disruption over the level over that entire time of that surge fill. And for this example, as I mentioned, it's about 10 hours for this process. If the PAT data that's being collected online is greater than the divert limit, the DCS will generate an alarm. The alarm will trigger somebody to come and observe and look at what's going on. And a magnitude and duration of the excursion will be used to determine whether divert is required before surge for this given process. In some cases, right, impurity control is achieved by implementation of divert limits upstream, right, of this particular unit operation. So there's multiple elements here that are being evaluated to understand and determine what the steps that we would need to take in the event of divert for this type of process. So detection, right, of an excursion, for this particular process is shown here. So busy slide, I'll try to walk you through it again. Analytical data that's being trended in real time along with our process data. And for this particular excursion, we detected roughly over a 4-hour period of time. We saw a spike in starting material 2 from less than 0.1%, which is typical of being monitored by NLC, jumped up to 1.9%, right? So should we divert this material, right? Should this impacted material be segregated and decoupled from the process? So shown here is magnitude of that material disturbance starting material 2, and this is the online HPLC data area percent being plotted here for that particular impurity. So as we talked about previously, we set this warning limit threshold at 0.5% for this particular PAT measurement, right? If the area percent of that impurity peak is below that, no actions required. We then set up control result limit, so again, HPLC area percent for this, where we set this divert limit at 1.50%, right? But we have this range where we can have either between, the warning limit of 0.5 and then below the divert limit, we're then exceeding that divert limit, which in that case, again, as I said, on alarm triggers, we go into triage mode of consulting our 10-hour rolling average model to determine whether or not diversion is needed for that particular material so that it doesn't impact downstream. And you can see the 10-hour rolling average model shown here in the black trace, where we have basically demonstrated that our process can tolerate approximately 0.26% starting material 2 in that pre-isolation surge at the end of this process that's collected. So as I mentioned previously, we're able to collect the different vessels in the surge over this time period, and we were able to go then pull samples from the surge tanks, transfer them to an analytical lab where HPLC area percent could be generated. And in all 3 of the vessels, both you can see before, during the disturbance, as well as after, the area percent of the starting material was well below the allowable threshold for it in this process, indicating that a surge or diversion of this material was not required in order for it to be forward processed and rejected using the crystallization downstream. So just to summarize and conclude some of the work that we did at this step, this was the first campaign that was run in this new facility in Ireland, where we had 3 chemical transformations. They were monitored by 3 dilution cards with 3 online LCs, and the results from the LC systems were able to be trended and dashboarded with our distributed control systems, and ultimately transferred and used to make decisions based on the disturbances that were shown in some of those examples. The results from that decision, ultimately, according to a strategy that we implemented for this process, right? That strategy may be different from other processes where we have different unit operations, different chemistry occurring, but overall, it enabled us to successfully demonstrate this process at a manufacturing scale. There's a ton of people to acknowledge here. I can't go through them all, but in the analytical team, Jeffrey Roberts, was a lead on this process. He just did an outstanding job putting together some of this strategy and partnering with some of our process chemists, Scott May and others in this. And I have 2.5 minutes, and I'll leave you time for questions. I just want to give you a snapshot of what PAT can do for another commercial scale process, right? So this is data for PAT that is used for convergent or hybrid type process for tetrapeptide manufacture at our facility in Kinsale, Ireland, where I don't have all the details here of the chemistry and the engineering unit operation, and I apologize for that, but I just wanted you to be able to see what PAT can do when it's deployed again in a controlled process. So online UHPLC was used in 3 different instances, monitoring this process across 3 different steps. You can see the number of samples that were analyzed, right? For chromatography purposes, that's an awful lot of samples being analyzed. And it's really informing us on the overall process health. We had 4 instrument problems over the duration of this work, over 4 months. You can see one was a degasser failure on an instrument, and we had 3 separate check valve failures over that period of time. Plans were installed and in place to address those types of disruptions when they occurred. Just 1 dilution card issue, so the sampling system feeds those LCs right at cracked O-ring, and it's something that's hard to kind of foresee, but overall pretty impressive performance of PAT for this process. Less than 1% of the PAT samples that generated results required human intervention to review. And, overall, just a good success story requiring only 2 QC analysts to maintain and oversee the operation of these 3 instruments in production. So, tip of the hat to a lot of the other folks on that acknowledgement slide for just some really remarkable work. Just to kind of go back, the photograph here shows the 3 instruments on the floor. You'll notice they're not ATEX rated, right? Because they're not in that hazardous environment, right? So we are sampling from the process, diluting and moving the sample away from the process so that we can run the instruments in an unprotected, in a safe environment that's still not requiring all the extra enclosure systems and controls. So with that, I'll thank you for your time, and I appreciate you giving me this opportunity just to share some of the exciting work that we've been doing at Willie. And, this photograph is over a decade of work by a lot of those scientists you saw in that previous acknowledgement slide. Chemists, engineers, automation scientists, quality control, QA, you name it. This is a total team effort, and we have the same ability that we're team really, and this project really exemplified that. So thank you.
Really nice talk. Matt Beaver at Amgen. Just a quick question on the control strategy. I guess the approach with the surge tanks. I couldn't quite see it on the slide, but the way you're describing it makes me think that there's maybe 2 surge tanks per whole point versus 1, what I would call a buffer tank. I'm just trying to think through the implications of those 2 different strategies and I guess how you decided on that particular approach.
Yeah, so the approach for this particular synthesis, right, was developed by our engineering team, right? And then the point here being, for at least, for the green buckets where we have this surge, right, is, it's twofold, right? So it's obviously, when you've got all the unit operations coupled, it's accumulating the product, right, that will ultimately feed in from the start, right? We're not taking material that was generated and then putting it into those tanks to allow each unit operation to start simultaneously, right? But secondly, it's also intentionally designed, right, to give us that ability, right? This was the first process that we ran, designing and developing, and putting it in this facility. We wanted to have an opportunity to potentially analyze things offline if needed in order to kind of preserve the process if there was a concept that couldn't be recovered.
I guess the specific operation, are you continuously feeding into the surge tank and continuously feeding outwards at discrete holes? And then once that's full, you then move to a different surge tank and then start to deplete the first.
So my memory is fading me here a little bit, but if I remember correctly, we are accumulating and feeding that and then pushing out to feed that next unit operation. But then, especially for step 4, right, we're collecting all of those tanks and then, those drums of material, and then each drum will be then subsequently pushed forward. So it's not a continuous push from a surge tank into the next unit operation for that particular unit operation.
Makes sense. Thanks.
Hi, Steve North from Continuous Pharmaceuticals. What unresolved challenges in continuous manufacturing are you currently working on?
So in synthetic molecules space, for small molecules, I would say, we're not currently, I would say, pursuing something new or not. I think, as we heard previous speaker space, some of these technologies and capabilities are pretty well established. They need to be utilized for a number of different chemistries. I would say, as we look at non-small molecules, synthetic species, there's a lot of opportunities in that space to grow, and develop and pursue ways to improve manufacturing. I think that's where I think if you follow the literature, right, or go to conferences and see a lot of what companies are being involved in, right, there's really opportunities to both take what we've learned and apply it to some new challenges that can potentially bring about.
Thank you.
Okay, so it's actually break time. We're gonna.
So, thank you, Todd. I really liked the talk. I love seeing the various applications. And so we're at our first break point. So if you would like to take about 15 minutes, we'll meet back here at 10:45. So thank you very much.
Is this going on in DC?
What's that?
Is this in DC right now?
Is that USB's headquarters? So I said to Jake earlier, maybe next year you can get 8% to send us down there.
Right. It's not too far away.
Because I think for students, I think it was something, I think it was only $100 to attend it.
It doesn't much at all. Do they have like opportunities for students to present posters or anything like that?
I don't think, it seemed like just a large group workshop, their sessions are pretty structured throughout.
Yeah, I don't know if the school has a policy if you're going to any conference you have to have some work presented. But it's not even like that expensive.
Right, that would be the only argument, it's not that expensive, and we could probably get a group rate if we were gonna take enough people. So, for anybody that came in after I mentioned it, the QR code at the top right is a website that they're using to take questions while the speakers are presenting. So if you have questions, just scan the QR code and it'll pull up the website and ask questions.
Pretty good. Only able to make it tomorrow, but at least
Just click that button.
Now it's on.
So hello everyone. I see there's still quite a bit of discussion as folks come back from the break. I did notice that despite it being 15 minutes, it went by really fast, at least for me. So I want to thank everyone for coming back. And we're going to head into our next session, next set of 3 presentations. And I'm particularly pleased with the next set of lineups. I think USP, when we went through the planning for this, tried to identify a good mix of voices throughout the industry, from generics through innovators as well as CDMOs, and that's what you're gonna hear in the next set. So first off, I'd like to invite up Dr. Shravita Ramakrishnan from Dr. Reddy's. She's Vice President Head of their API manufacturing group. So, come on up.
Thank you very much, and yeah, thanks to Gabriela for inviting us to talk at this forum. So I'll be talking a bit about what we have been doing in the area of continuous manufacturing. We are still in our journey in implementing this, and what are some of the challenges that we have faced, and where we see our path forward. So so basically we are about a $3 billion generic pharmaceutical manufacturing company. And our distinctive, strengths that we look at are, we have a global presence. And the reason I'm talking about this is because, end of the day, it also ties into why we are interested in continuous manufacturing. And one of the main, aspects for us is affordability. So, being a generic manufacturer, we do want to look at the cost of both the drug substance as well as drug product. We are vertically integrated, though we offer drug substance to external formulators as well. And having robust manufacturing operations and productivity is very important to us. And we also look at patient-centric innovation that's relevant here. So in the generic industry, the context is a little different from the innovator. The cost competitiveness is very, very intense. Many of the APIs are already almost commoditized. We were discussing with Stephen over breakfast this morning on metformin. You're saying how in metformin, like almost everything that can get squeezed out of the cost is almost there. Though there are still maybe some potential advantages to be found. So there is an intense competition in traditional generics. And we also want to get into more complex, injectables and biologics. And there is also a lot of disruption through digital. So as we talk about AI, how does it impact advanced manufacturing? And these are all, various factors that are changing around us. So sustainability is one of the main areas of focus for us, and we have various sustainability goals, across from reducing carbon emissions, making our products accessible and affordable to patients. So the affordability is a major criteria. We also look at equity, diversity, and inclusion. In fact, I have an additional role also as a chief diversity officer, and basically the highest standards of compliance and ethics. So once again, affordability, innovation, sustainability, all form a major part of our organizational goals and objectives. And this has actually led us into flow chemistry and continuous manufacturing. And there has been a growing interest in this field. I call this article now, this is a 2018 OPRA article that shows the various number of publications on various unit operations. I'm sure there's something more updated and recent now, but we couldn't find anything that actually summarizes how many publications there are. But one thing you will notice is reactions is a lot, crystallization some, you get to further downstream operations, filtration and drying, there's really not much out there, right? It's still a very much evolving field. And many advantages of flow, which I won't get into since it's being talked about, but while the advantages are manifest, there are still certain challenges that we face. So our journey in flow actually started about 8 years back, where we built a small platform for handling cryogenic reactions, right? And this is mainly for the beauty, chemistry, and this is just for just for that step of the whole process, and the rest of it would be in batch. So it's kind of a hybrid process. And then in 2018, we were actually challenged by our chairman to say, can we look at a modular plant, right? Modular continuous plant. And really, I'd say it was his vision at that point in time that got us seriously thinking about a much larger scale for continuous manufacturing, right? And as we looked into it, we decided to pick up one of our flagship molecules. And again, this was the start of our journey. There wasn't much out there. And we thought it might make sense to pick a molecule that is high volume for us, right? And this molecule is about, maybe 100 tons or even more than that per annum. So, which is pretty large for an API. And we decided to pick this molecule and, develop a continuous process for it. So we started that journey around 2018-19. The pandemic kind of slowed us down for various reasons, even, with getting various equipment on time and even progressing our work. But there were additional challenges as well beyond the pandemic. And right now we are in the process of commissioning our plant, and we intend to validate this process by the end of this year, right? And one of the major things that came up, and I'll describe a little bit about this process because it talks about the whole context of how we got into it. So there are 3 reactions there, and in 2018 we were pretty fast. There was no major chemistry challenge. We were pretty quick in establishing the POC in the lab, and within a couple of months we even ran some pilot scale trials. And we said, oh yeah, we can do this in flow, we can really intensify the reactions, find a single solvent system, make it more efficient. And we were then challenged by our management to say, well, it's fine you're doing reactions, but what's the point? Because downstream, at that point in time, they were doing it in batch and drying it in vacuum tray dryers. So you can imagine a huge tonnage molecule. Imagine the number of vacuum tray dryers they were using, and later they did upgrade the drying technology to other dryers such as filter dryers, water dryers, and so on. But still very huge, large dryers. So the challenge to us was, if you're going to do this molecule, do it all the way, right? Do it all the way to drying. And being, I would say, completely naive in this, and we kind of jumped, I would say, into the challenge without quite realizing what we were jumping into, and said, okay, fine, we'll look at trying. And it was in 2018, I went to Akima, found this nice crystallizer, and then, searched for a dryer, and we said, okay, yeah, here are all the technologies. All we have to do is kind of, set them one after the other, and then we are done, right? It's quite simple and straightforward. So that was where we were in 2018, right? And I'm sorry, I think there is, so I think the order of slides is slightly mixed up for some reason, but it's fine. So that's kind of, I'll just skip through this because I already talked about it. Yeah. So we said, okay, we'll do all the way through. We started with the 3 reactions, and you can see here the time. Is there a pointer or something? This one? Oops. Okay, I'll just use this one. So in batch, you could see the reactions were on the scale, took several hours because you're intentionally slowing down the reactions in batch. In flow, we could really intensify it to just seconds and minutes. And there was a big challenge around the byproduct filtration, which I won't talk too much about here, but we'll mention it briefly. And then we got into crystallization. So in crystallization, we really could not intensify it because crystallization requires that time, right? So we didn't really see an advantage of doing crystallization in a continuous mode, except that it kind of links the whole train. And then we got into continuous filtration, delumping, and drying. And for drying, we could, really do it very efficiently with the continuous fluid bed drying. Yeah, so it's just again in the order of minutes, we could reduce the water content. So the final crystallization is sort of IPA water, and then you have a water wash. So we could reduce the water content from about 50% down to around below 5%, which is like the stoichiometric hydrate, in just a matter of 12 to 15 minutes. So very, very efficient drying. Right? So we said, okay, all the operations are nice, efficient, and we ended up designing this whole continuous plant where we had, we selected these silicon carbide plant-based reactors for the first reaction because it was with hydrochloric acid, highly acidic at 95 degrees. And silicon carbide was the only material that was actually compatible with at this temperature. And then we had a divert point here to check the pH, and if the pH was still acidic, it would divert because we didn't want acidic pH solution getting into the plug flow reactor here. So we had a plug flow reactor just to extend the residence time once we added sodium hydroxide here, because adding more plates into the tankrix would really increase the pressure drop and make it really unviable. So even here we had 2 tankrix in parallel because if we had used only one, the pressure drop would have been too high. So beyond the PFR, we then had, so happy to hear the last speaker talk so much about the water spatral UPLC. So we had this UPLC here. And one of the challenges that we have faced is actually getting the UPLC to talk to the control system, because Waters kind of said, okay, it's very difficult, and they were really not able to help us much with it, and we suddenly realized we had to figure this out on our own. So, so these were kind of the very unexpected challenges. We thought we had it figured with the UPLC, but connecting it to the control system, having it talk to each other was a bit of a challenge, but I'll take some tips from the God's talk and see what we can do better on that. So, and then we had a surge vessel here. So there is a divert point and then the surge vessel, following which we then proceed to the next, the next reaction, which is a sodium to a calcium salt conversion. And once again, the divert point here before it gets into this byproduct filter. So there's a slimy mass of calcium hydroxide. And this is where I think converting something from batch to continuous becomes very challenging. People talk a lot about reactions, but hardly anyone talks about byproduct filtration. And doing that in continuous is not easy. You say in a batch process, yeah, I have high flow bed, you prepare it, you can filter it through, and so on. But doing it in continuous, just synchronizing these two, and then there's some time gap. So what do we do in between? And we have a supersaturated solution coming right from this reaction. It's supersaturated, so we do not want it to precipitate till it enters the crystallizer with the seeds, right? Because if it precipitates before it enters the crystallizer, then the form is going to change. So there's a lot about solid form control here, and the crystallization part I think is an entire topic on its own. So, but I just say it is very, very challenging to control the solid form, the particle size, and we had 3 CSTRs in series. We had an RTR and 3 CSTRs because we really needed this RTR to control the form. It's very interesting, but if we don't have this, we go directly to crystallization in CSTRs. Over a period of time, we see the amorphous content start to increase and we lose the form control. So that's an interesting phenomenon that happens there. And then into filtration and drying. So when we said we'll do this, what we didn't realize is what a challenge it can be to really balance this entire set of equipment train and make sure if the crystallization slows down, then the filtration, if the crystallization has fines and the filtration slows down, then that impacts everything all the way back. right? So having this kind of very precisely balanced train is really, I'd say, something we didn't anticipate in the very beginning. I think the order slides is really messed up for some reason. I'm sorry, but I'm going to have to skip this. Yeah. So when we went into this whole thing, while we had the vision from our chairman, we looked into the so we implemented. The capital investment ended up being more than our initial estimate, right? And rationalizing this return on investment was quite a bit of a challenge, And we also find that in general there is a lot of sunk capital, which makes it difficult to justify the investment into a continuous plan if batch is working just as fine. And just to expand on that, like in this particular case, we invested around $14 million, right? We looked at for a capacity of about 100 tons per annum. But when we actually compare the raw material cost of the batch process, the difference is negligible. While the operating expenses, the overheads operating expenses are better for flow, we still find that the difference is still negligible. If you look at actually a return on investment and the payback period of 22 years, it doesn't really warrant a need to invest in this for this particular product. Yeah. So, and I'll come to why we're still doing it in the next step, but just going further into challenges on this. There's also a need for multi-purpose plants. So when we had this CapEx approved, it was with the condition that it would be multi-purpose. Now and this is essential for a generic because we don't want to have a line dedicated to one product. But that also makes it a lot more challenging because when you're talking about this kind of scale, which is 100 tons per annum, making it multi-purpose is not so easy. So I think the reactors part of it is still, we can switch it around, but it's much more difficult to switch it around than we had initially anticipated because of various we can of course modify piping, but it's not like a 2-day switchover is all I want to say. It is possible, but not a 2-day switchover. Right? And for crystallizers, filter, dryer, that's all set in place. I mean, it will obviously follow that sequence, but the process also has to fit those particular crystallizers, filters, and dryers. We found expertise on advanced manufacturing technologies again to be a challenge, right? We were starting from scratch, learning pretty much everything, to a large extent on our own. And the more unit operations we have, the more complexity there is. And there's also, while we talk about academia industry collaborations, and we have, while we try to collaborate with, academia and experts across the globe, I think that's still something that's evolving. And for academia to really, understand the challenges that an industry faces, and the speed at which we have to develop can be pretty good. Yeah, and evolving publications, I'd say, is another area on challenges and failures. So what we find is, while in this case there was no chemistry challenge, but when we've done some beta lithium chemistries or some other chemistries, we actually find that even the reactions that form impurities are accelerated. So you're not only accelerating product formation, we also see sometimes a worse impurity profile than we do in batch. While everyone talks about how flow gives you an advantage in terms of purity and all of that selectivity, I think we're not seeing enough publications where it doesn't give an advantage, and how do you actually overcome it? So I think it's important to see a lot of these failures and understand how to overcome those as well. So, I'm sorry, I don't know why the slides are up. Yeah. I think there's something that's getting mixed up with this. It's just randomly swapping the slides. Do you mind if I connect my laptop, because I am completely lost with the flow now? Excuse me for a minute. It's just, I can just connect this one to the laptop, right? To my laptop. Will it work? I think the orders just totally switched. Yeah, absolutely.
switch it back there. Just a second. There you go, see how it flashed? So now, I gotta go back into the back and switch it. Come on, you see?
Okay, that's fine. So in the interest of time, I'll just continue on this. Sorry about that. So in the SWOT analysis, when you looked at, these are the strengths, but weaknesses we found are in downstream operations, in the crystallization, filtration, drying, where how do you handle fouling and crystallization, and it's not been attempted much out there. And the complexity of the control system, when you have so many operations in series, we have like 2,000 IOs, and all of these talking to each other. So how do you handle such a complex control system and knowledge required for it? And there are also limited equipment manufacturers, which is leading to a high cost of equipment. So that further raises the challenge to invest in this. The opportunities, of course, are in safer and green processes, and with minimal manual intervention, we can get greater consistency in the process, right? But the threats are, I'd say, really a business case, which I talked about, and a mindset shift, right? So even we found many people viewing this as one complex project, rather than really an organizational change to adopt advanced manufacturing. So you need a mindset shift across the organization and people buying into this. And the last is the scarcity of talent and expertise, which we really find there are very few people who know this, and and those who know it, like there are companies just trying to kind of learn from each other and move forward on this. So we talked about this, and it's fun. So given the challenges in business case, what we really want to look at is the holistic assessment of benefits. So I showed that in terms of raw material cost or this, we really don't have a payback. But if you do look at the production time, the amount of QC load, right, analysis that goes, and I just made an estimate that a batch would be like for 3 days of run. It could be longer, of course, right? But you have a reduction in number of samples that come for analysis. And the green metrics are still better. Yeah, so it's a fairly significant difference even in the green metrics. And these are aspects that, even though if we look at only a business case from the investment and payback, it may not always work out. But looking at other metrics, can be very beneficial. And sustainability, and that's why I was talking about sustainability in the beginning. Given that it's one of our major objectives, we find that this is significantly beneficial to get into continuous manufacturing. And we're now continuing our journey, screening a pipeline of molecules, right? Where we're looking at handling hazardous reagents, greener routes where we have lower solvent consumption or a cleaner synthesis which could not be possible in batch. And identify those novel process windows where flow can offer a significant advantage over batch. And more efficient technologies even for extraction, distillation, and so on, which will help us integrate everything together much better. So the ICHP 13 has really, been great in terms of giving us a holistic view of what we need to do. But there are still some questions which I would say, as we are working on this and we're getting towards our performance qualification and filing the DMF for this product that we have. So while there's a flexibility around batch size, we are curious how should a validation batch size be with regard to, say, a commercial batch size, because we're not sure in commercial how long we'll run, how much flexibility would we have there, right? And so what should be the duration of the run that's established in process validation as against commercial manufacturing? Or for cleaning validation, like some of the plant-rich reactors, we can't really open them up, right? So it's got to be written samples, and is that good enough? Process dynamics, there's a lot about residence time distribution and all of that. When we have so many unit operations that are all linked up, it's not quite easy to do, and especially when you, at the end of this for the crystallization, we have CSTRs that are holding like 2 kL or 3 kL volumes of the slurry mux, right? Because it's a huge scale. So establishing residence time distribution, and that would be total residence time for the crystallization is about 9 hours. So imagine having to establish that and the amount of solvent that we would consume to actually do that in real time with solvents. So is CFD good enough to estimate a residence time in such situations? And how do you link up for so many operations, the residence time all the way through to drying? So the technical complexity of this is, I'd say it's still something we're working on. And again, I talked about multi-product facility. What are the quality requirements for product changeovers when you're looking at flow equipment? Yeah. And in terms of sampling, so we collect material in multiple lots, but when you finally call multiple lots a batch, then what is exactly the sampling that will be required, to say that this is a batch? And control strategy, the stuff on material traceability that we look at that the guideline mentions, ISH 230 mentions. However, do we have to do material traceability for every reactor, right? Do I do it only for my main starting material, or do you have to do material traceability for everything that's coming, the hydrochloric acid, NaOH, whatever else, right? So how much, to what extent do we have to establish material traceability? And that again becomes very complex with so many operations in sequence. And during a validation, for example, should we demonstrate the system's capability to handle disturbances, right? So intentionally maybe have a disturbance there and see how it reacts. And if a batch, for some reason, has multiple diversions, does it need to be placed on stability? So these are just an example of questions that came to us for which we couldn't quite find the answers. Maybe it is still there somewhere, but we couldn't really, find the answers to this. And further, in terms of practical guidance, I'd say, while a lot of the what we need to do is well addressed, how to do it is not very clear. So I'll just give one case study as an example. So while we were running this during some of our trials, this is the flow rate of hydrochloric acid, which is very steady. We have these Fuji triple piston pumps. The flow rate is absolutely steady. It's wonderful, right? And then there was sudden aberration for a few seconds. So the limits we have here, so it's exactly at the set point. We have a high and a low defined, which is based on our design space, right? And beyond the high or below the low, it will trigger an alarm. And then there's a high and a low limit where we say we'll divert, right? And if it actually exceeds beyond that and goes to what we see are the plant shutdown, PSH or PS low limits, it'll actually trigger a plant shutdown immediately, whereas it says it's gone way beyond the limit for whatever reason. And in this case, as an aberration just for a few seconds, it actually hit the plant shutdown level, but then it recovered and then it actually stabilized itself. But by that time, the shutdown got triggered. Now, we might have just kind of been overcautious in our approach in stating that it should trigger a shutdown as soon as it hits the plant shutdown level. Should it really, or can it wait? So how do we actually address questions like this? And while there's, a lot of, while the ISH 230 defines very nicely how to use process dynamics, RTD, and so on, like actually how we go about, doing it in a real-time situation. So here what we did was, we looked at the reaction kinetics. I'll just give you one example on this. Reaction kinetics for this particular reaction with HCl, right? And we validated the model. This is in the lab. And again, when we talk about models and kinetics, what we found another challenge to be was, you have to control this particular starting material down to below 0.1%. So the model has to have the sensitivity and accuracy to predict below 0.1% if I'm really going to use this for any further, model-based diversion strategies. And getting a model down to that level of sensitivity and accuracy is not easy. It took us months and months of experimentation to get it to be so precise. Yeah. So typically in the batch, you say, oh yeah, my product is consumed. Just look at the overall kinetics. That's very easy to do in a few days. But getting it down to 0.1% of a starting material is really tough. And then we had established the RTD models, and I'm just showing lab equipment as an example. Fed all this into a G-prompts model and generated a funnel graph. Yeah, so it's a dynamic simulation which shows the various components during the reaction through the G-prompts model. And in the sensitivity analysis, we probe each parameter and what will be the effect that it has on, say, a specification limit. Right? So we find that, say, if I vary x1 by 5%, it doesn't have much impact on y, but if I vary x2 by 5%, it significantly has an impact on y. So it's more sensitive to x2. And we generated these funnel plots. So for example, if there's a disturbance in the HCl flow rate, right, it has an impact on the intermediate formation, and when the disturbance lasts, the x-axis is the duration, say about 40, yeah, here, sorry. So when it lasts about 40 seconds, it actually hits this line where the spec is below 0.4% for this intermediate. So we through these funnel plots, we know that, and this is for NaOH, we generate a table like this that says, okay, if this flow rate varies by say plus or minus 15%, which of the specs does it impact? It doesn't impact the starting. In this case, for example, HCl, it impacts the intermediate formation, and it can tolerate only 40 seconds of this disturbance. Yeah, whereas it doesn't really impact the starting material or rather the impurity. So we pick which is critical in each case, and that then determines the time required for the diversion, and couple it in with RTD and the logic to say this is what the diversion strategy should be. Yeah. So this is kind of how we went through the whole thing. But the question always arises, like, are there some good case studies which we could refer to know, can this approach be improved? Can it be better? Right? How do we develop further on it? Right? Because this is our own thought process. And while I've attended conferences and I see a lot of talks on chemistry, and I've seen a few people talk at a very theoretical level about what should be done. Very few talks or even I think publications on practical examples of how do you go about something like this. So this would really help us. So in how do we actually decide when to divert, what should be the duration of the disturbance before we decide to divert, and so on. So just finally, accelerating adoption, I'd say workshops on the regulatory perspectives would be very helpful, and FAQs for practical implementation. So glad to hear now that we have more and more submissions and approvals. So if there can be some success stories that are published, right? That say how did people implement some of these things? I think that would be really helpful for us to quickly learn and go forward. And some consortiums of public-private partnerships that include the agency, academia, and pharmaceutical companies, including generic, would also really help us to learn quickly and adopt this technology much faster. And of course, thank you for talking about approvals in the morning, so that was on my list here. Yeah. So thank you, and I think there's a huge team. This is half the team that's in the lab that's working a lot on this, and there's a bunch of folks in the plant who are running these studies day in and day out, and doing a fantastic job in ironing out one issue after the other. And it's been a series, I'd say. The huge learning is always the unexpected. So I was sharing an example very quickly this morning about how we thought we planned everything. We anticipate power shutdowns in India because by the time the backup comes on, there'll be a power interruption. We had everything on UPS, and then when the power shutdown actually happened, the instrument air was the one that went off. Right, all the pumps, everything was running, the process was fine, but the instrument air went low, and the sensors started tripping. So this is the journey of the unexpected, I would say. Yeah, thank you.
So thank you very much. I really appreciate your talk. I think we'll hold questions for the panel discussion, and I will ask Ernie if you would come back and bring there you go.
Sorry.
Oh. So up next, I'd like to welcome down to the stage Dr. Olivier Decremont from Ampac Fine Chemicals, who's going to go through a presentation about the modular design aspects that you're working with. So, thank you.
Thank you very much. Thank you very much for the invitation and giving me the opportunity to talk about this development we've done in the last few years here at Ampac Fine Chemicals, and address some specific issues on scale.
There you go. Give me 1 second. Just say public.
All right, it's a long title. It shows up, but it's basically the development of a technology to help us facilitate the scale up from the bench top in between before going to commercial scale. There's a gap there, a gap that unfortunately takes too much time to fill right now, especially at this pace we are developing processes. So we're going to address this today. So when you talk about batch process development, this is a typical life cycle for a process. You start early on with the chemistry that comes from, discovery, not very fit for scale up. And as you go through the different phases of development, the chemistry increase the performance stepwise. Steps are more or less bigger than others, but as you go down the path of the phase for the supplying clinical material, you improve the chemistry, your knowledge of the chemistry increases. Eventually you get to commercial scale, you still need to continue improving your chemistry, your process, refining your process to get better, more cost effective, because now you have generated pressure, you need to maintain the market. So this is a very typical progression, and it helps because when you do this, you're going to invest a lot of money up front on a drug that might not make it to the market, right? Phase A, phase 1, there's going to be a lot of attrition at this point, so you don't want to have a lot of money in the game yet. When you get to commercial scale, you need to maintain the knowledge and improve the process. So there's this very typical, shouldn't be a surprise. Obviously, you hope that the price goes down, $1 per kilo. Small scale, we never talk about the $1 per kilo. Manufacturing, we talk about the campaign price, because otherwise it's a ridiculous number. It scares more and more people. But most optimists know that if you pay $200,000 per kilo at very small scale, you should be less than $2,000 or $3,000 a kilo at commercial. And you all have those scale down process in your head. But when we talk about continuous process, it's a little different game, because it's not just the chemistry. Now you have to have a lot of chemical engineering in place. So you need to know the chemistry. You need to develop a chemistry that actually is amenable for continuous process. So the batch reactor can take a lot of things, a lot of mistakes, a lot of uncertainties in the process. When you go into continuous, you can't have that. We need to put a lot more pressure on the chemist to know the process way up front, to have a better understanding of the mass transfer, heat transfer, kinetics. So you design your reactor, or you use a reactor that's going to match the process and get the product that you need. The beauty of this is that if you do all the work up front, you don't need to have a lot more optimization later on. But again, you're starting to do a lot more investment up front where the drug may not make it. So this is the conundrum that we have in this industry. So there's the rate of attrition of drugs so high, it's very difficult to spend a lot of money up front with a drug that has a high possibility of not making it, right? So how do you address this? Well, you need to have equipment that's available. You need to have rural scale up. You need to have automation that's appropriate for your process. And of course, we talked about PAT, so you need to have all those tools. Fortunately, in the past 15 years, a lot of tools came available at small scale for the chemists to develop chemistry with a lot more ease than we used to. We have all those micro reactors that allows you to do very fast screening of conditions, reaction kinetics. And by the way, that's the hardest thing to ask a chemist, is to measure kinetics. They hate doing that. They like to throw things in the reactor, walk away, come back, and then hopefully the reaction's done. How long does it take for the reaction? I don't know, overnight, it's done. Continuous process doesn't work, because you're not going to design a tube that's 2 miles long to handle that resistance time. You need to have a tube that's a few feet at best, and high temperature, high pressure, right? So you can't do that at pressure. So you have to optimize your chemistry where. Good. So yeah, from a CDMO perspective, it's very difficult to develop a continuous process because we don't necessarily have all the tools. In the batch chemistry, you have all your multi-purpose plants. We all have our 10 liters to 2,000 gallon reactors available, so you can do the scale up, and you all know your rural scale up from going for phase 1, phase 2, phase 3, etc. You can do that in generic equipment, and you can continue, and it's just a question of choosing a few pipes to organize your train the way you need for your chemistry. When you do flow chemistry, it's usually flow process specific. So now we see a lot of people developing chemistry very, very efficiently, and then they have to build a system to run this chemistry. COVID came and put even more problems in that, but providing flow meters, electronics, PLCs, that kind of things takes a long time. Building a skid that's developed specifically for your process takes a long time. We have customers telling us we need to get in phase 2 by end of 2024, when you have basically just a bare bones for the process right now. It's not going to be possible to get the equipment in time, right? So you have to find a solution in between. You need to adapt the chemistry to the existing equipment. Fortunately, there's not much equipment existing here. So there's a significant capital investment right now because we have to build a system. So this is the ideal thing. You do your process characterization at the beginning. You go into the bench top. Bench top is just HPLC pumps, tubes that you get in the drawers, oven, things like that. Very easy to set up. And then you have to go to the demonstration scale and the process to make your phase 1 and phase 2 material. A few words on PAT. We touched that early this morning. Early on, in the early phase, we want to do as much PAT as we can. You need to measure everything you need in your process. And then later on, you figure out, well, I don't really need to measure this. Why can't I measure that? It's easy to measure, like a flow, like a temperature, like a pressure, because those are very easy to measure, instead of being something extremely complicated to measure that when I have a signal that to deconvolute and take a few hours to get the results back. There's some solution with HPLC systems, that Claude mentioned, but again, you still see something that's already occurred in the past. So by the time you get your result, even if you can get it within 5 minutes, this 5 minutes really might have been produce something that's not in spec, right? So how do you react to that? So hopefully, by knowing your process, the more you know your process, the least things you need to measure. The example of continuous chromatography, we do a lot at AFC. We do chiral separations. We don't have a polarimeter online. We could, be great, but we don't have to. We can take a sample and do an HPLC offline, because the dynamic of the process is slow enough that if we are steady state, if something fails, it has to be a dramatic failure, and we'll see that with the flow meter, the pressure, or the temperature of the system. All other evolutions will be slow. It's a slow decay of the performance of the column. The torsion column is going to lose performance over time, but it's not overnight. It's over 2 months, 3 months. So you can actually monitor that very easily. And you don't have to rush a sample. You take a sample, take a sample every 8 hours. You take a sample whenever you make something that's might affect the outcome. You have a new feed coming into the system. You have to make a new solution of solvent or XYZ, and you need to measure that impact over time. So you need to measure resistance time in the process, etc. So the easier, the more you know your process, the less PAT you should have, because the more data you have to handle, the more difficult it is to make a decision. What happens if data is conflicting? Think about it. That's actually a problem that the petroleum industry is facing every day. There are people with PhDs on that. When you have too many pieces of information that are conflicting, which one is the one that you need? So when you develop a process, obviously the chemistry still is in the process there. You still need to know your chemistry. You need to know better the chemistry than just mixing A and B gave C. You need to know, do we degas something? Do we make a salt in our process, right? So you need a chemical engineer that's there to say, hey, I need a balanced equation. I need to see where the CH3 is going. I need to see where that oxygen that's there, that unseen on the right side here, what's happening? Do you generate water? Are you at high temperature, high pressure? When you go past your BPR, that pressure regulator, what's going to be the solubility of all your stuff in there? Are you going to still be a solution? Because now you're not at 20 bar, or you are atmospheric, right? All those things, chemists don't care. I batch, because it's just a stir reactor. The chemical engineer is going to come and tell you that. Then you have an electrical chemist that's going to tell you if what you're making is right, or what you can measure to control your process. And then to control your process, you have an automation guy who's going to tell you, well, that pump is not really the right pump for your process, because you're telling me you need a flow rate that's plus or minus 1%, and the pump that you're using is a peristaltic pump. First of all, it's not going to get to the pressure that you need, and the flow is not going to be stable and that kind of thing. So you need all those people to get together. We saw that earlier with the presentation from Lily. It's a community effort. It takes a village to raise the kid, right? This is the village. You have whole teams is here. But then you need to evaluate other things. The core of the process is the chemistry, but then you have to run the chemistry for a long time. You're going to consume a lot of solvent, you're going to consume a lot of material. If your rate is 100 mils a minute and your bottle is only a liter, you're going to have to refill that bottle of 1 liter very frequently. So take a bottle that's 10 liters. So you don't have to refill it more frequently. And then your process is going to be more stable because 10 liters is a constant concentration that's in there. Every time you change a bottle or you change the raw material, you're going to have a difference, a small difference. Hopefully it's not important to your process because your process is robust. But that takes into consideration. You have cycle times in your process, your batch size. Usually the batch size is defined by your individual batch process that's at the end of your synthesis. You have an isolation in a dryer, most likely it's going to be a batch process. How much material you put in that dryer is going to drive on how long you're going to run your process to define your batch size. Your quality control, obviously, you don't want to do testing every 5 minutes. You don't want to have a batch every day because then you have some batch records to review. You don't want to have a batch every 6 months because then there are a lot of materials at risk, right? So you have to find all those balances. All those things need to be taken into consideration. And that's when we're talking about GMP. Obviously, event management, we had a lot of conversation about when do you decide to divert your flow or not. The best way is to have the process as robust as possible. That takes away some of the performance of your process. That takes away some productivity, but it makes your life a lot easier if your process is very robust and you can accept a small change or a large change in some of the reagents in your process. You don't have to worry about it. Your downstream is going to take care of it. Your chemistry is going to be fine with it. That will have to affect how fast you can run in the system, what is your productivity. And eventually you need to have some control strategies and automation in your process, which takes me to the transition from benchtop to GMP manufacturing. Well, we got an R&D lab, we talked about small scale volumes, throughput, we made 500 grams of material a day. It's already pretty good, pretty decent. This is done on the bench with PLC pumps, oven, things like that. This is typically your chemist. It's an 8 to 12 days run. They don't run overnight, they don't run for 24/7. It's not GMP, so you use lab notebook, your cowboy, 25 liter cowboy, that kind of things. 10 mils a minute, that's typical flow rate, that's an HPLC pump flow rate. It's fewer hundred liters in a week, that's not a big deal. You go to the right side on the commercial scale, you want to be over 50 kilo a day, because you're going to look at metric ton quantities. This is a system that's going to be running for a long time. You have to be robust, you have to have full automation in the system, so very enhanced system control with decision trees with AI maybe to help you make those decisions, or a subject matter expert that's in-house. You need obviously validated systems and huge storage. You're going to still go through thousands of gallons of solvent, thousands of gallons of your raw materials, and then you need to be able to handle the downstream process. But typically that's what we do in commercial scale. But the key is in the middle here. The 5 kilo a day range. That's what you need for your clinical trial. You need 20 kilo for clinical trial, run for 4 days, you get your material. Before you run for 4 days, you need to have a system that's validated because it's CGMP. If it's phase 1, you still need to have IQ, PQ of your equipment. It doesn't have necessarily to be validated, but you have to have some control system. You have to have some batch records to run. It has to be fairly robust. You're talking about much larger volume. So this is the size that's critical for this industry because this is what drives the speed to market and the quality of the process. And you need obviously a process that's scalable from the bench to commercial through that GMP kilo scale. So you can simplify your process using a modular approach. So on the top right hand, you have a process view from a chemical engineering perspective. You have in orange is your streams coming in. The purple is your heaters or heat exchangers to cool or heat the solutions are going into your reactors, or EHG in red. And then H is of maybe a back pressure regulator or a heat exchanger to cool down. But most of the process can actually be driven like this. You can take a lot of processes and they will look like that. There may be another stream coming in somewhere, but they will all look like that. very, very similar. So from a chemical engineering perspective, all of this is just unit operations. So we could do the same thing in the automation side. So the digital piece is that you have your modules on the right side, your pumps, your liquid extractor, your PFR, all your modules are on the right side. And those are your modules that you qualify individually. Like any piece of equipment in the batch plant, your reactor is qualified. You installed it, you qualified it, you qualified its functionality. You don't qualify it every single time you don't use it. It's qualified, it's in the system. Then you have to have the PLC, which is the brain of the system. That's the one that can gather all the data from all those modules. You need to make sense out of them to control the process. That's where you have your feedback loop for pressure, for flow rate, pressure rate, etc. So in between you have your I/O, your interface, and then you have a historian to save all that data. You have a compiler, right? You need to have the data stored, not modifiable, and you can actually retrieve it. And then you have your interface with the operators, your HMI, down there. So all those pieces individually can be all IQ'd and PQ'd and validated for every process. So that approach, what it does, it provides you flexibility because you can change things and you only have to qualify the ensemble, validate the ensemble, right? So you can put another pump in that system, you just need to make sure that the pump can communicate with your PLC. That's where the trick is. And then when you do this, you can build a library of modules. We have quite a library of things. We have different pumps. You can build them in the same frame with the same design specification. That way they can fit together. It will be like Lego blocks. You can assemble. There's one I/O line that comes out of this module, and then you can put them all together this way. So you can do your system qualification. The beauty of this is that if you go to the next process, you just need to replace maybe a couple of things on the right side, those small modules, but all your PLC historian I/O is the same, and then you just adapt the HMI. The HMI, if you have a system that's well designed, it will take only a couple of days to get it modified in the HMI. So as you plug your modules to your PLC, you should be able to recognize what's plugged, and you can assemble your system very easily. So that provides you a tremendous amount of flexibility because you can go from product ACC 616, that's how we name our product, ACC is our code for our product. You can go from the 616 to XX product or YYY in a very simple amount of time. Because your automation system is already in place, you just need to validate or qualify the small changes you have to make. And all the modules are there existing already pre-qualified, so you just need to put them together. You have a recipe for every product that way, because you have one instruction that tells you how the modules have to be assembled, what modules are assembled, how you assemble them, and then how you qualify your assembly by itself. You make a modification to any of the modules, you have to document that through change control, like in quality systems for any batch reactor or any piece of equipment at the plant. So that allows you to have a lot more flexibility. So you build your process. I'm going to skip this, and we're going to go an example. This is a 4-step synthesis that we implemented using this technology. I'm not a chemist, so I'm not going to put the structures in there, but this is how I see the process as a chemical engineer. Those are my unit operation. This is only step 3 and 4, and all the steps. So you define your process like this. You get the data. As a good chemical engineer, you need the kinetics. You need to be able to model your data, and you can see underneath the reaction model. You have water, gas, and water liquid. It's part of the model. You need to have that because you're going to be operating at high pressure, high temperature. You need to understand what's happening in your system. And then you need to do what we call a deep dive analysis, so FMEA on your process. You need to affect the probability of an event to occur. What are the chances that my back pressure regulator is going to fail? What's the impact of that back pressure regulator failure on my process? Why can't I detect that? And you put a number on those things and you calculate what that number is. If the number is very big, you're in big trouble. You need to do everything you can to minimize that number. If the probability of my BPR to fail is high, and I find a different BPR, different manufacturer? Can I find another way to control the pressure? And if I can't find another piece of equipment, can I put system in place that I can detect when the pressure fails and have a backup to that? Right? Those are kind of things. So you do that analysis, deep dive in the process to really understand what's happening and find how to mitigate those risks. And then you have your modules here. So all the little squares are the piece that will be represented by physically a piece of equipment. And then you go to your module library and you put them together. And they are, you can see the size of the modules, they're pretty small. On the top picture left, they're about 2 feet by 2 feet by 2 feet cubes. You assemble them together and you can see a picture of the HMI, it's very simple. In this case, we were starting from a very small tank scale because that was just for the demonstration run, but now the equipment has been moved to another location, which also helps because it's very small equipment, it can move very easily. It took a day just to go across the US, a couple of days, and we installed within a week. And then you can see that all the equipment fits nicely in the. So, an example on the fly change. Really quick, this is the same chemistry, step 1 and 2. We had no problem at small scale, on the bench scale, but then when we went to that kilo scale, we started to see blockage and it was a problem. So we had to analyze what was going on. We found out that the amine was actually making a salt that was not a problem at small scale because of the piping was big enough to let that salt go through and we didn't really see it. At a larger scale, the ratio of the pipe diameter to the flow to the parameters of the process was such that we had a point where solids will accumulate and eventually plug the flow. So without changing too much the process, this is the setup. The step 1, step 2 were 2 reactors. The second reactor was removed and got closer to step 1 reactor. This was actually ambient. And that allows very simple modification to the process to keep going within a day or 2. That shows how flexible the system can be and how you can adapt. And you can still see problems if you spend a lot of time developing your phone. Rapid implementation, real quick. This is something we see a lot with the power separation. We do a lot of rotary isomer separation. The good thing is the undesired isomer usually can be racemized very easily by heat. So you just push it through a tube, 180 degrees, 200 degrees. You have to cool down before you go through your BPR because you don't want to go through the BPR very hot. Because you might have some issues on the other side. So it's a very simple setup. And this is the module setup. You can see the flow at the bottom, the pump, the reactor, a hot temperature reactor on top. Pretty big, it's a coil inside that tube, and then the cooling tube, and then the back pressure regulator. And that was set up within a week, and we ran 3 batches already on that process. That's the kinetics of the reaction, racemization. So we can see how much resistance time we need and so forth. Energy production here, you can see the gauge almost 200 degrees on the first reactor and 50 degrees on the second reactor. Pretty simple setup. This setup was done without even automation. So it's just a pump, you just put out in the pump, so it was PLC type pump for the flowing we were considering. Demonstrated at that scale with 5 kilo a day. That's a nice material. Conclusion, we need tools to go through clinical trials material faster because that's the key of this industry. We need to go commercial fast. Used to be 10, 15 years to get a product from discovery to commercial. It's less than five years in certain customers. Obviously it depends on what kind of clinical trial you have to do, but the indication. But the faster we can get to the market, the better, and the faster we can eliminate the drugs, the candidate, because it fails people time, the faster we can get, right? So having the tools, like those tools I showed, to be able to rapidly get process A into the plant, make the 20 kilo, 100 kilo material that you need under CGMP with the quality systems in place, and then pivot to another project within a week or so, is key for a CDMO for like us. Some of the modules can be dedicated to the product, so you don't have to worry too much about contamination or excessive cleaning. You can dedicate some of those modules because they are fairly cheap, fairly small. It's only one piece of equipment that you have to make. You don't have to make the whole skid. And if one module does not exist, you don't have to build again a full skid. It's a small piece of equipment that you have to build. So within a few months, weeks, you can build it if you have some parts already on the shelf. Flow meters, pipes, sweatshop fittings, that kind of things. The cost is going to be tremendously lower because you don't have to pay for raw equipment. You will pay just for that specific piece of equipment. The rest is in the library, just a question of putting them together. You don't have to pay for a brand new plant every time you put a product in a batch facility. You pay for the time of the plant, you pay for the amortization of the plant, but you pay for the brand new plant every single time. From the time you go to GMP, small scale, you shouldn't have to pay for the full plant every single time. And with that, thank you very much for your attention. If you have any questions.
We'll hold questions again on this one, just to make sure we stay on our time. So thank you so much. So the last presentation before lunch, I'd like to invite Dr. Eric Jajak, who is the Director of Process Engineering at Vertex Pharmaceutical, up. And thank you very much for coming in, Eric, and take it away.
Okay, thank you for the opportunity to speak today. This is something I enjoy speaking about. Today we're going to go away from, well, right now we're going to go away from drug substance towards the drug product. I have to say up front that this is not just about Vertex, but it's kind of about my career trajectory from one of the original large molecule, big pharma CM innovators through to working in CDMO and working on bringing technology there to now back with an owner with Vertex. So there are things in here that are controversial with my company. So somewhat my personal viewpoint. My talk today is going to be broken down to three parts. One for powders, why continuous manufacturing is better. I'm not going to focus on regulatory aspects of business or anything like that. I can bore you with that another time. This is really about why it's technologically better. Why is it a better mousetrap? Why does it produce better product? Then hopefully I establish that, and then the question is, okay, so you got a process, it's better. Why is it not being updated? Like I said, it's less than 1% of material are going into humans right now is coming out of the CM line. Why is that? It's been 10 years. And third, I'm going to talk about where I see a direction that could go to eventually make it beneficial for companies to go in this direction. So, not to belay the point, why is continuous better? If I could take everything and boil it down into one statement, the statement is superior blending. What do I mean by that? These horizontal blenders that we see in the process, they are fantastic at blending powder. It was one of these things that 15 years ago when we started looking at this, Ditya in the audience did a lot of research on this in their thesis. This was the number one question, was how are you going to know the blend is good? And when we started testing it, what we found was the blends just work. It's more about feeding. You have to pay all the attention to feeding, and that was really our learning a couple of years ago. So this is in conjunction to batch, where what you have in batch, and there is a picture, if you can see it, that's a room size V blender. It's a 1,200 kilogram scale V blender, where the idea is that in continuous, our primary means of scaling it up is to run longer, and then you can also scale up by going to larger throughputs. In batch, we make the process bigger at each step along the way. So why is continuous better? Well, in continuous, when you have those paddles, they're going and they're sweeping through the powder, and they're really working on scale scrutiny that we care about. We care about what's going on with the powder down at the tablet type of mass level, right? And if you go to a running longer, it's the same process. If you go to a bigger process, what we actually find is that when you go faster, Your feeding gets better, because that's your feeding is loss of weight feed, there's uncertainty with that. If your feeding gets better, you need less back mixing. So there's even a means of scaling up where you're not worried, where you get very similar environments of blending, even when you go faster. So even if you want to go 10 kgs an hour to 100 kgs an hour, there's a very nice pathway that I've certainly seen means that you don't see a different product out the other door. Compare this to the batch equivalent, which is we start at 1 kg, go to 3 kgs, 30 kgs, 300 kgs, maybe 1,000. Every time we do this, it's a very different environment inside of that blender. You start to see a whole bunch of things like a lot of mechanical forces are much more intense within that blender. And so what we see is that we have processes that react very differently at batch to at development all the way up to commercial scale. And we know this, we see this all the time. The other thing is that our scale scrutiny, we go to a bigger mixer, and now we still want to then blend down to this 1-inch level. So you need to put more mixing time in to get down to there when you're blending this small volume level. But while you're doing that, you're putting more shear, more power into it, and you're getting a different product out the other end. So basically, what Continuous offers you is the ability that in development, you can study your process, and you can study your process that will be indicative of your commercial process. What we do in batch to this day is we can't run independent experiments on 300 or 150 or 600 kilograms of material. So if I was going to run 30 experimental points in the batch world to understand multivariate and see how all my different factors and formulations work together in order to control experiments, let's do that. Let's just say 20 points in the DOE, 20 points in the DOE at 300 kilograms, multiply the cost of a kilogram of drug product, and you're up into the hundreds of thousands, if not millions, if not tens of millions of dollars in material. No one studies their commercial process in blending on a full QBD basis. We study it down at the 10 kg scale, 30 kg scale, and then we take a guess. We jump right up commercial, and then we just run 3 batches. And if it passes, we're good and we go. If it doesn't, we play with it until we get 3 more. We really don't have statistical power to understand what's happening at our commercial scale. This is where Continuous gives you a pathway that is better, and it's going to ensure that you're going to have better quality throughout your life cycle. So that is it. That's why it's better. It's because you get to focus the blending. There's a pathway for scaling. You're not worried about having a different process when you scale up this with this blender involved. All right. So if you believe me, it's better. Let's talk about why things aren't being taken up faster. And this is a tougher. This is where we start to get into like the cultural thing, and thing that honestly at this point I'm kind of bearish in my career about whether or not this will ever be uptaken by the on the drug product side. So the first one is that in the last 30 years there's been a ton of investment in OSD capacity, and right now what we have is we have a glut of capacity available, and that's worldwide. That's in the United States. That's in foreign markets. And so what we see now is that when you're competing, you're not competing against the new capital cost in Continuous. You're competing against an existing, already depreciated piece of equipment that is free to that company. That's driving the cost down there. It's not a apples to apples. So there's less incentive to install to invest in this. But then the second part, and this is something that I think some of the early innovators feel, is that right now there's a lack of outsourced manufacturing capability. So if you do produce something in Continuous in-house, your only option for business continuity is really to convert it to batch. So then that comes with all its own regulatory hurdles and problems. This is probably the least of the technical challenges. I'm going to kind of go in what I see as ascending order. The next one, and this one's really fascinating, it's been fascinating my whole career, is having to do with the pharmaceutical model for process engineering. So particularly in drug, not so much in drug substance, somewhat in drug substance, but definitely in drug product. Like what's happened over the last 70 years is that the equipment hasn't evolved or changed very much. And so as a result, pharmaceutical companies have said, hey, we don't really care to innovate on a tablet press or a granulator. We're going to let the vendors do that. We're just going to design the facilities and drop the equipment in the room. So what you see now is you see that we don't have engineering in the way that most other manufacturing companies, most other manufacturing industries really would. What do I mean? This is the design process for pharma. We start off, we might have business requirements. I've actually been on plenty of projects that don't even have business requirements. It's just, we're gonna install a tablet press there because we know we need it. Then you go through and do a conceptual design. Sometimes, a lot of times that gets combined with basic design in order to go faster because you know you wanna put the piece of equipment in the room. Then you go through basic design, you start figuring out what particular vendor you're gonna use or whatever. And then you go through detailed design where you start figuring out all the things that you have to do in validation and qualification. Straight through, straight shot. This is what I want to do. I'm going to buy this equipment. I'm going to put it in the room. Works great when you're installing tablet presses. Let's talk about most other manufacturing companies. If you just Google engineering design process, like this or some form of it will come up. This is not a straight line. This is a loop. This is number one, what's the opportunity? What's the business? What am I really trying to solve? Number two, let's do some research on this. Let's actually go take data, figure out what's going on. We take the data, we understand what's going on. We figure out how that can actually be used. We prototype things, we test. We go around this loop until we have a high degree of certainty that we're going to successfully execute on the program. So again, this is a type of thing that, like, if you're going in a direction you haven't gone before, this is a requirement in a way. What I think is most interesting about this is that a lot of times I hear people say, oh, well, we need the projects to go quick because we need to have a good payback on it. This is the food companies and the chemical companies that are making pennies per kilogram of margin. They invest in this not because it's more expensive, but because they can alleviate their risk by doing the cheap stuff, the R&D up front, and then put their major investment later in the project so that is where they can start receiving feedback from fast. Again, I've never been in a pharma area where like we're gonna do a 5-year or a 10-year project. That's how these other companies survive in these other places. Okay, third one. This one's the biggest, in my opinion, that I've seen in the drug product side. So, again, continuous process. I've been a part of four or so continuous manufacturing processes. All of them were pretty much Six Sigma plus capable in blending, without a doubt. Everyone was rock solid when you look at the blending. However, there's also been this expectation that they all have a real-time probe in the process, and that's a requirement in order to show that they are actually still blending capable. That's a real mind-boggler for me, because like if I go and look at the batch side, we know that the number that's usually put out there is 2.5 Sigma capable as an industry, and it's definitely, blending is definitely the Achilles heel of the industry. When we look at our process capability, we start to look at things with content uniformity and dissolution. Those are usually blending related issues that come out to the quality side. But there's no real-time orthogonal expectation of measuring in the batch side, right? So there's this thing there where we have a higher capability, but yet at the same time, we have a higher onus to demonstrate it within a regulated environment. So, bring on the third part, what needs to be addressed first. So in my proposal here is that the pharma manufacturing system should really be tuned so that the testing burden, the burden of how much and how intensely you're monitoring and testing your process should be inversely proportional to the capability of your process. If you can demonstrate through your program your high capability, you can also then define how you're monitoring and showing your quality. This is something that right now is kind of like a flat level across all products, and I want to say flat. So what does this look like? How do we do this? Batch and continuous are inherently different, right? So how do we do it? Well, if I've learned one thing from working in the continuous manufacturing space, it's that there's a ton of value that comes from looking at your process as it's running in real time. What does a sample at one time point? It gives you a great deal of information about what's happening at that one time, right? And at the end of the day, if I'm making a tablet or I'm making a capsule, at the back end of that line, there's a tablet press or an encapsulator that's cranking out a continuous flow of product, right? And this is where the two processes are the same, and there's an onus for understanding what the capability of the two processes are. And what I'm going to say by that is that we have to start thinking about process capability not from the way that it is defined everywhere within the pharma thing. So this is an example. Let's just say we're looking at the content of a tablet, and we're running. And as we're running, we take in this case 5 locations of tablets, and we go up to the tablet press and we pull. Each time, I don't know, I think I did 10 tablets in each pool. Do 10 tablets, go through, HPLC them, or at line near IR, whatever you're gonna do, and get your distribution. 10 minutes later, do the next. 10 minutes later, do the next. 10 minutes later, do the next. At the end of an hour's worth of time, you go back and you look, and what you see here that, there's some variation to it as far as how much variability it has, and there's also some variation of how much it's moving around from the mean, right? So you have your variability, which is really drawn out, if you know process capability, by your CP. This is how wide your distribution of product is compared to your specification limits. And then there's your CPK, which says, okay, this is how wide it is, but you know what? It's not on center. It's cheating towards my limits. This means I'm going to have a higher probability of a reject. And to control, both are important, right? I need to both maintain what my capability is, but I need to keep it near the mean. And we don't do this in a batch. What we do in a batch is we say, oh, it's a batch. So we expect the blending is going to be uniform. So we're just going to sample randomly at the end, losing that time information. And when we sample randomly at the end, we're going to look at what the performance of the batch is overall. And that can be seen by what we have over on our right, which is going to be really like what the traditional industry would call a process performance or a process performance capability. So if you look there, there's no way to distinguish whether or not you have a problem with your variability or you have a problem with your mean moving over time. So let's look at a different process where this is one where I ran a different simulation, and this is where I said, this one's a much less capable process. It's a much higher mean, but it's much more centered on what's going on. You can see that these two processes here have very different fingerprints as far as what you do, how do you attack them, where the problem is. This one, you need to figure out why you have variations, what's happening. This one, you need to figure out why your variations, so why. But if you look at them from a batch perspective, just pulling a random sample across the batch, they look pretty much the same. There's no difference, they're the same capability. So this is one of these things now that there's no reason for us to say, I'm gonna randomly sample a batch because it's a batch. Pull the tablets in time and get the information. Then you could go in and somebody could come up and say, how's your process running? And say, oh, our process has a CP of 3.1, and we have a PP of 2.2, and somebody could very quickly say, oh, that's a good, you're in good control there, right? You're doing well. You could probably improve your overall control a bit more. So this is one of these things that, again, the way that pharma deals with it is pharma says, I'm going to measure my performance of the whole batch. One batch, I'm going to randomly sample it. That's going to tell me what my instantaneous capability is, my CP and my CPK. But everywhere else, who will manufacture out of something like a tablet press, they will pull multiple time points during a thing, and each of those time points will represent an individual CP or an individual CPK. When they combine them together, then they can understand the process capability. And the big thing here is that means that within one batch, you understand what your process capability is. What is it in the standard? We say you need 20 to 30 batches before you understand the process performance in the batch world, because we're assuming that a batch is all of uniform quality that fits to a Gaussian distribution, which is not the case when you stay in the prototype. What could this mean, in the life cycle? Well, this is where companies could build in a control strategy around their filing, where like during development, you'd be at level 1, which would mean that while you're producing, maybe every 10, maybe you'd take 10 locations from your batch while you're producing, and you would take 10 tablets at each, and that would be level 1. If your capability is above X, then maybe you could certify yourself into going to level 2. Where you're going to only 3 points. And then after you see so much experience within that, you could go to level 3. And you could go through the different schemes of testing until you get down to the point that maybe you're testing one batch every 6 months. If you're running it all the time and have really good capability, you're going to be taking a risk. If you find a batch that's off, you're going to have to investigate that and look all the way back. So there's a business question in there. But you are doing this based on on how you're demonstrating the process in real time. And again, there will be criteria at each level. So for instance, like, if your CP is greater than 2, or you start to see a change, then your system would say, hey, we just saw a major change in our process. We need to come back down and reestablish this baseline. If we need to go back to level 2, we need to go back to level 1. So benefits. I think one of the big discontinuities is that in the batch, we tumble the blender and then we pull it out and we sample it and we think that's what it is. We don't have a lot of means of understanding what's happening. In continuous, we have these feeders that are telling us what happened. We have RTD models. We have all of these additional means of understanding the capability. So that type of information also dovetails into this nicely, because when you're going through levels, you're going to talk about what the risk is that you are not understanding what the risk is that you're missing a change. And here you have this other rich thing of data. So this should enable you, having all this extra information should enable you to A, show your capability quicker, but then also accelerate through your testing levels quicker, because you have a higher assurance of quality. And then overall, be able to demonstrate your control within a regulatory framework. All right, so conclusions. I think understanding dynamic conditions in both batch and continuous is like the key to really understanding and putting out there what type of, how to set up logical, evidence, capability-based understanding of our processes. Again, I think this is key, that continuous should be ultimately cheaper because you should be able to run it with a higher confidence, and it should be able to be something that's tested less frequently and we have higher confidence in. But in order to do that, we need to change the way that we think about the whole manufacturing process batch included. And then finally, while this is not maybe super for all CQAs, I'm thinking about like the solution here. There is a majority of CQAs and a majority of products, probably 90% of products out there, or 90% of the CQAs that this would be good for, that would ultimately be able to give a means so that more capable process, be they batch or continuous, have a more economically viable path for manufacturing. Because honestly, right now, for a lot of companies' perspectives, having a couple batch failures because you got unlucky in the way that you sampled, it's not that big a detractor economically, and there's not a reason for them to invest in the time, talent, and technology it takes to build continuous. So that's all I have. Okay, I got us back on track, a little bit back on track. Yep. Sure. Yeah. Any questions? No. Hungry. Thank you.
So thank you everyone for your patience. Now it's time for the lunch break. Lunch will be right out here. You can go into either the meeting rooms. If you'd like to go outside, you're welcome to go outside into the courtyards, kind of brave some of the Canadian smoke that has come down to the area recently. So thank you.
daylight.
Yeah.
Yeah, it would definitely be interesting to go in person, which we would do now.
Is it in Canada this time?
No, it's down in DC. Yeah. They mentioned the wild parts though, because I think it was New York City and DC have like the two worst air qualities in the country today. Wherever
That's
it's
why they
from.
mentioned Canada.
Yeah.
About the fire.
Yep. I noticed it yesterday. At one point, I was looking outside and you could look right at the sun. It was just an orange circle in the sky. Very interesting.
Today there was a lot of smoke, like it wasn't clear.
It's hard to tell because it's usually so gray around here. It's not unusually cloudy. table. Do you need help? I'll grab it. You say that or yeah? I'll grab it. I'll grab it.
Just Tasha, head out.
We just need a quote.
No, no, that's perfect.
We also have a lunch tomorrow.
For what?
Because the
Oh, for
program
this?
is done before noon.
This?
So just breakfast, right?
Yeah.
I like the perspective. I love events like this. But it's a way to start. Has anybody asked any questions using that thing?
I think Natasha might have.
I've asked 3 questions. The first one they deleted. I'm not sure why, it wasn't controversial. And the second 2 are waiting for authorization.
Let your voice be heard.
I think continuous manufacturing is it's.
You should ask the same question. We should all submit the same question.
We should. What was your first question? We can all submit that question. It was basically, it was the guy talking about, it was Adam Fisher, the guy from NTA. Oh, yeah. He mentioned that there were 50 approvals into the emerging technology program. And that there were 20 approvals from that. And so my question was, what approval are you talking about? Like a approved drug products? And if that's the case, is there a published list of drug products somewhere? Because like for the first couple of years, there was like, there are at least like 7 different articles that say, here's all the here's all the approved products. The last one of those, the last one's from like 2019. The question
Oh,
is,
help yourself. Help yourself. Only.
What's the problem? That is a hard question.
That's a really good question. I can't believe they wouldn't respond to that.
So I think I asked it again in different wording. It is a difficult question. Because when they ask it, the guy, it's not that way. Oh, I don't want you to appreciate. I said, was the sample sizes in your self-audit, superior results?
We will start self-search. We will start self-search. Once the CO2 is connected, CO2 with our self-delivered state from too late. That's for we will check the efficacy, sensitivity, and we will just ask tell them that this was effective with this cell regarding against the C4, C5, and they will grab the cell to the mouse. PDX, right? It's PDX, right? That's interesting. That's just the practice. Our culture is Zenkumer cell. Human mouse is on its way, and human is we have.
That's always a nerve-wracking question for a state. Just say yes. It's true. You have the long history.
I'm sitting here today. I'm sitting here today, not in the lab.
What?
Listening. They have a workshop. It's a workshop. USP. Dr. Linden is not coming today. I talked to her over the phone and I confirmed she's not there. And I said, I'm sorry. No more today. We have a lot of homework, of course. But she will come tomorrow. 10, 12, and 1 appointment. Do you have like a template or like example of the proposal, NIH proposal? Like we have to write the method part for her. We are a little like assessing her writing, but she wants us to write a method. And it's method. I try to. For our home, she said this is for your home. Like preparing went to like 4 points. R41. Choose a part. Then we have none. Otherwise, and we have 11 candidates. A lot of research teams, but we have 11. I think for some of the part we are working on, we have 11, 14 targets. 11 or 12, which means a lot of work. Each one, I think each one is for the master's column, right? Master's amount. Then if we combine 5, then it's like a PhD. It's just like a 2 PhD or 3 PhD. Or could we have just watch it? I don't know. When do we move? August.
What is it?
Hmm?
What is it?
This one, I think, this one pepperoni.
What is this?
Oh, this one is orange. Do you want some orange? No, it's not. I can pour it and then give it. No, it's okay.
There is orange juice for it.
That's your favorite mandarin. Banana.
Cherry juice or anything like that.
No
There is
juice.
something but something else.
That's why I brought water. You want some juice? And then we split. Look at this. I can drink 4 ounce. You drink 5 ounce.
They encourage conference attendance for PhD students, for PhD.
So how much you are learning? How much you are learning?
From a lab?
There was like
Lab?
mandatory like social
Oh my god, it's totally different.
diversity training, which all of the diverse people I
It's
went
like,
like about,
how you enjoy
which
it?
I was like, yes.
Because I learned Western blotting, not just like why we do this, and then she explained those things very well. Like not like a method or technique, like a big picture, like why we choose this, for example, like, and then what's the something like always like everything is relevant, useful, not any single thing is not any waste of my time. And she's very dedicated. It's like it's like method. It's like she made you until you understand. And she like it's like teaching you like until you reach her expectation. I think doctor will be 100 times higher than me. Dr. Lick's standard will be 100 times higher than me. Oh, how did you said you emailed to your committee. Can you forward the one, like just a copy the comment, not the forward, just a copy the sentence? Because I have to say in person, so I have to put it to the committee.
I can directly give you forward the email.
But not forward, just to copy the sentence. Not forward, that's not good. Not good. I mean, what I'm saying is, you know what I mean? Like just to click
The
forward.
wordings.
What I'm saying is that I'll change it, I change everything, but I'll like
Just to get an idea. Get it.
like because I have to say in person, so I have to ask all the people sitting there. Regardless the discipline.
Just a note.
Okay. Oh, sure, I have a banana. Oh.
Yeah, I appreciate it.
But like, that's like.
Thank you. Yeah, we're all charged up.
I think it's way more helpful, especially for, especially right now.
How about you? How is your life? How is your life? Good.
Actually better.
Better. Better.
I feel like it's so. We're all interacting.
You will be closer to my boss in the same neighborhood.
I hope she doesn't know that.
She doesn't know. I mean, in terms of like, she doesn't care about the gossip. She's like a think big. She doesn't care any gossip. She's not interested in the gossip. She could like notice, but she doesn't say anything. She doesn't like make any kinds of stuff. But that's a little bit public, but for also a little bit like a friendly. We talk a lot, but it's not like a gossip. But we talk a lot. We hear what's going on with her. You know what I mean? And then her research, and then she explains a lot of things about how she designs or how she thinks what's going on. Like it's like a learning life. Because usually they may not tell anything like just do this, just do this. But she explained, oh, we do this because we we are doing this.
I used 3 of the slides, and one of them was just because I couldn't remember the exact definition.
We are not writing actual writing, but we are at least learning some part, which is good. Which is good.
Yeah. Absolutely.
When your new student is gonna come, when is your new student gonna come? First.
8 or 9 hours.
Nice.
You guys won't have any.
No.
Only 2 of us.
That's me. She has the one student and those. That's why I couldn't change it. That was really perfect time because she asked one student to Dr. Anderson, and I was talking to Dr. Anderson. Dr. Anderson said, oh, she said, and then did you talk? And then I said no, but I love to go.
And what happened? Actually, Dr. Anderson gave you I want to go there.
I didn't say, oh, I wanna go. I mean, I didn't say like we spoke each other, but I said, oh, I spoke my things in that lab with Dr. Rittenelby because I could trust her, and then she's so wonderful. And if I can, I said to Dr. Rittenelby, like, oh, if I can go to the lab, then that would be perfect. So I appealed to him. Dr. Anderson they didn't say anything, and then Dr. Rittenelby didn't tell me like, oh, I love you or something like that, but I communicated with Dr. Rittenelby, like I said, oh, I met Dr. Anderson and I discussed like this. I kept gave a signal, like appealed in dialect. appeared, and then I said like, oh, this is how it's going, and then something like that. Because she didn't say yes or no, so I was like, what do I say? I have to stop the, I have to stop the carbon now, something like other labs, or other even discovery, right?
Right to all of them.
I have to do.
But you didn't.
No, because
You were already selected.
when I first spoke with doctor, the doctor said, oh, that's perfect match. She's looking for, and then you go, you can go. That was his plan.
What was the option if Dr. Veterinary was not available?
That was, I mean, I didn't know it's available or not, but why?
Yeah, I'm asking what would have been your option?
If it's not available, then when I spoke with Oxygen, Oksana at the time, also other graduates, senior students, they said, oh, you have to start with job because they expected students at that time. So they said, try to like narrow down the professors, even in other discipline as well. But I was hoping, if not, then maybe Dr. Carbon, because your professor and Dr. Roden is, they know with other professor, previous professor, so that may not like affect you. You know, like oh, there is high chance they won't accept it.
That's not the case. Because there was a student which Dr. Johnson like didn't want to continue with. Same as your case, like
okay, the professor,
didn't want to continue with that student.
What happened?
And that student was given to Dr. Lee. Dr. Lee took her. And
That's nice.
She completed her PhD. Even though Dr. Lee and Dr. Johnson were very good people, they were very like Dr. Johnson and Dr. Lee shared a lab as well. It was very similar to Dr. Lord and Dr. Lee lab. It was similar to Dr. Johnson.
Got it.
So he took it.
So which means your advice is nice and generous and thoughtful. And
what I'm saying is he assumed that
if it happened, that is that good or bad?
he
Better
assumed,
or
he assumed that she might not take the student or a person who is
then
like,
I was gonna speak with other people as well, but thank God.
so there was an option for other subjects as well, like other departments as well.
So basically, Dr. Anderson asked, do you think your previous advisor will jeopardize you? So I said, if that's professional, of course no. That's why I couldn't be in the same concentration. Otherwise, I have to really find out. Then he will find out. But I said, but I feel like a threatening. Recently, I feel like a threatening, but I don't mind. Because I'll record everything just to send to school. Send to at the company.
I'm happy you stayed in the concentration. I'm happy you stayed in the concentration.
Of course. So while I was studying in my undergrad, pharmaceutics was popular. We call it like a pharmaceutics is like a flyover of a pharmacy. But another like a major part is college, pharmacology.
Because they have an option for pharmacology.
Oh, you are only pharmacology?
Only pharmacology.
Oh, that's nice.
There's no chance I can go in. Because the problem is my background is pharmacy.
Oh no, it's not. Pharmacy is like pharmacy is like you learn how you formulate.
No, no, I'm saying that my undergrad was of tea farm or pharmacy. So that's why I don't have that much of idea of pharmaceutics or medical chemistry. So
Before us, we had a very brilliant professor who studied pharmaceutics, and he was
you want to eat more? I want to. I'm bringing more.
third round.
You heard about the latest on the cookie? I didn't know that. Oh, so they have decided that they cannot possibly find the That's oh, I heard that. Yeah. Like if they close the gates, it's no longer are used to carry illegal drugs.
So we had a very nice option because the pharmaceutics professor went to Michigan and was in a pharmaceutical company, and it's like a really like a brand new. So that was a good option for the pharmaceutics, but for the pharmacology, it's like a really like a tradition, like old fashioned. But later on, I found many of my colleagues they studied pharmacology, and it's wonderful. You know, it's like a core. It's like a core, but pharmaceutics is like for the company. I don't know. It's like more like a not I don't think it's more like an industry. But pharmacology might be a little bit like academia. Like a research way, different kinds of research. But
Right, and that's the problem. Everybody's working in tandem, but nobody's working together. So like, we're making progress, but we're not even close to, getting any efficiency from calibrating. You know? I mean, actually, okay, where they're actually using this, which is the problem. Like, I feel like nobody goes into the literature.
I'm having pastime today. I'm having pastime writing here and then read email and just watching that one and also checking email, answer the phone, pick up the sales. Tomorrow is it will be okay because yes. I'm also doing. I did yesterday a little bit in Epsilon, where I made some, and then I'll do rest of them until Thursday, then combine everything, then submit it to the
It's true.
and we will staying with Ellie Jelly with oil wet for ourselves later. So, but not like a fancy microscope, but we will take any take images later.
The B is also under microscope.
Use your microscope?
To be has gotten
I know. I know Olympus.
I actually do not.
I know Olympus. I know.
She came to me on Saturday. No, I was asking, can you guide me with that?
We try to learn.
It's brand new.
Brand new, it doesn't mean anything. You know, experience is important.
Yeah.
I also used, I didn't tell you, but I also used the computer, fluorescence microscope, fluorescence microscope. We had other microscope as well. My previous experience is we made the microscope in the lab.
You made microscope?
We combined it. So in that case, we took out this lamp and we combined everything, checked everything. But that was it's like I don't know, it's like that one. Also for the computer, I had to go to other school. And then I brought myself to another professor. He did it because that's literally he did this kind of like microscope also, and then it's very expensive. So but it's like all the techniques are like experience, even though there's the what is it like the protocol or steps. So you have a fancy microscope and you have a fancy Odyssey.
I don't have fancy microscope. We don't have fancy microscope.
It's fancier than us. I think we share microscope. But for me, I don't care because I know how to do that. But I don't care. But I'll just rely on our.
From the day when I came over, I realized that you were in a lab which will make you do PhD in 6-7 years. Because
our first lab?
Yeah, because
10 years. 10 years. Not
most of the time you would be working for the lab more rather than yourself. But now I can confidently say that if everything goes smooth, you will be out of here in 5 years.
Because Dr. has a plan. Every year she has a plan for the student as well. So she gives us like a paper. She gives us like information, research in, and then she gives and she also guide us, and then we keep changing things, and we collect the data, and then it's not 100% like a plan, but we still, she's giving us this is my plan for you and you and you, so and then it's like a lot of guide.
From the first day, I thought that you won't be clearing PhD soon.
20 years. I think 20 years. We will buy, I would buy the 5 machines for 10 years, then doing experiment for 5 years, and then collecting the data must come from the company in 5 years. 20 years.
I also told to Vivek that Myount will take a lot more than us to clear PhD. He said yes, but he would be able to learn how to develop a lab.
That's not good. I think that's not good.
Yeah.
That's not good.
First off.
It's not like that.
Yeah. Totally.
I have to finish early.
Totally.
We have 2 minutes, and that session will start. Unless you wanna hear.
I don't want to have any shaking.
Okay.
I will just go.
Okay. You can feel free to ask.
Are you done?
Yes. Thank you. Thank you. Water. For your thirst.
Bye.
Thank you so much.
Have a good session.
Thank you. Thank you.
I hope everyone had a good lunch and some time to maybe step outside or do some other things during that lunch period. We're going to move into our next session, but before I do that, I'd like to introduce Denise from RAPS, who is a co-sponsor of our event, just to say a few words, and then we'll move on.
Hi, good afternoon. Thank you for coming out today. I especially enjoyed the presentations this morning, a lot of really great information. I'm looking forward to hearing more. RAPS is pleased to co-sponsor this event with USP. RAPS, for those who do not know, is the Regulatory Affairs Professionals Society. We provide education, professional development, and membership for folks in the regulatory space, in pharma, biotech, med tech, as well as the folks who work regulatory adjacent in the same industries. I've got a little booth outside with some information about our group. If you have any questions, I'd be happy to answer them. I won't be here for the whole meeting. I do have a couple other conflicts, so I did leave my business card out there. There's also some flyers and stuff about the programs that we offer. Thanks for coming out. Welcome to the meeting.
All right, thank you, Denise. And so next, with regard to our case studies, I'd like to introduce Frank Witolski, Director of Engineering for Merck, who's going to come up and give us another fantastic talk. So, Frank, thank you.
And I just want to congratulate USP for having tacos up there for Taco Day. Like, if that was an accident, I don't know. So yeah, so I'm going to talk about another drug product case. So sorry to keep you with drug product after, but thanks USB for the invitation and audience for listening here. So I'm going to talk about Merck's first fully continuous drug product process, and we had a bit of a novel control strategy. So Eric was talking about, why do we need to do that product testing? Can we just use an RTD model? In our case, we did. We didn't get worldwide approval, but we at least got FDA approval. So that's what we're going to talk about. So in terms of our outline for the talk, I'm going to talk a little bit about our approach to continuous manufacturing and the business case and product selection that we used. Then I'm going to talk about the control strategy for that product and with that focus on the novelty around the RTD model without NAR, which is very commonly used. I'll talk a little bit about our regulatory interactions, and then finally talk a little bit about where we are today. So starting off there with our approach. So why did we consider CM? You've heard a lot about that from different people this morning, so I don't need to go into it too far. For us, the focus really was on flexibility to meet product demand. We have all these processes where you have this huge piece of batch equipment and it's designed for a really large batch size, and then towards the end of product life, the demand goes down and that's too big. Or we're developing a product. We developed a product for treatment of COVID or something. We had no idea we were going to be selling billions of tablets or barely any. You didn't know. So if you can design a process that has that flexibility of batch sizes, you can meet your needs so much better. So that was really our focus. And other people have talked about some of the other benefits. The product that we worked on was Januvia. So we took a different approach from some of the other companies. Instead of using an NCE that we were kind of tied to with this technology, we took an existing batch process that we had over a decade of data with, had developed using QBD, already had real-time release. So we took that and we used that as a base case to convert that over to a CM process, allowing us to really play with that continuous compression process without that sort of business pressure. You're not going to launch this NCE if you don't get this approved. So let us kind of move a little slower and really optimize what we want before we go in with a new chemical. And then, sorry, I did not advance the screen because I'm using two computers. And so then, in terms of what comes next, so we're now installing a high potency capable continuous direct compression unit. Unfortunately, the one that we were working on that I'm talking about here was spun off to Organon when Organon separated from Merck. So we don't have that equipment, but we will have a new one. It's in production now, and that will be a high potency because our current pipeline really is that. We're also doing a lot of development work in other technologies like granulation. So I think that's something that's really important, is to be able to utilize all kinds of technologies, not just direct compression. Although we do think most products will fit for continuous compression. So just to give you a little more detail again on that pilot product we used, we had been manufacturing it commercially for over a decade. It had a high overall volume, over 1 billion tablets per year, but that volume also did depend on the dose. We actually had some very small volume images that were only for Japan, and then our lowest dose, even worldwide, had a pretty low demand. So that gave us kind of the opportunity within one product to work with some sort of larger batch sizes as well as some smaller batch sizes. The product itself also was pretty easy technically, I think, from a CDC perspective. The drug load was over 25%. That allows us to use weight uniformity for dose uniformity. It was BCS class 1, which kind of minimizes some of your bridging concerns. The film coat was non-functional. The product itself is low segregation potential and also relatively low stability risks. So we didn't make it hard for ourselves. Secondly, we really wanted to sort of demonstrate and develop the process that we wanted without some of the risks that we deal with some of it's a little more difficult. Although we've certainly done a lot more in the development space, and honestly, I talked about how good the vendors are and things like that. The technical concerns really aren't there. And then in terms of the process, so again, we did utilize tablet NAR spectroscopy for assay, but that was 3 tablets every 15 minutes. It really wasn't representative of the, I should say it wasn't representative, but it wasn't showing you that second-to-second control of the process. The bulk of the control, the assurance, was the design of the process itself, a really good denominator mentioned, as well as using the RTD-based process model to look at the feeder data and ensure that the product quality was consistent throughout the batch. As I mentioned, we could use time of weight and VOCU. We didn't have dissolution testing for this product or aeration testing, so that was relatively simple. So here is what the facility looks like, and I think the first point to make is it's pretty small. So it's around a third of the size of what we would use to make the same amount of material in the batch process. So that is pretty important. I guess I should say what we're seeing. So this is the GIA CDC 50 line on the right, and this is the twin consigma coders also from GIA on the left. So what you don't see is above this where you feed the material down into the system, but on this floor, like that is all manufacturing equipment right there for the 1 billion tablet a year design. Our goal was to have less than a 90-day lead time from when we started formulating to getting that product to the patient. And again, we're really focusing on that flexible batch size and meeting product demand. So I wanted to talk a little bit about how we designed our process to ensure that we were doing it. So I titled it there, beginning with limit line. So again, starting out by using an existing product allowed us to sort of minimize the risk of delays. We talked about the design. One of the things we really worked on with design, we heard that some of the early installations of FAN had long changeover time because of the high number of changeover parts and the difficulty of cleaning. Our upper management challenged us with the phrase lightning fast changeovers. We wanted to get things down to a shift or less to go between images of a product, and maybe around a day to go between products. And we did demonstrate that sort of we never got to commercial production, but we demonstrated that in practice. One of the things we did, a couple of the speakers mentioned the importance of cross-functional collaboration. So this wasn't us in development land sort of working in our ivory tower without working with the people who are going to be making the product. We had really close integration of the manufacturing staff, including an operator who was with us who were live in design, technicians who were there, quality was there. We all worked together, talked about what we wanted, talked to the vendor about what we wanted, and really worked through that all together cross-functionally. I don't think this is the kind of thing you can just do in a silo. And then finally, in terms of the software integration, again, we heard that some of the earliest installations had some issues around that, so we really worked closely with the vendor and with the site on that. We recognized the need for some talent build at the manufacturing site, and we made that happen. Okay, so here is a overview of the equipment that we use. So it's a little hard to see, but that is where we charge the raw materials into IBCs. In the case of the lubricants, there's actually smaller containers that we're able to see there. Actually, you can see the little white containers there. And then the IBCs hold the larger volume excipients and the drug. Down on the first floor, we have the CDC 50 itself, which is the the blenders integrated in with the tablet press. We used a Bruker tandem for tablet NAR testing as well as weight thickness hardness. The coders, and you can see on the floor right there is the solution prep tank. And then something unique that we designed with the vendor is called the distribution arm. So this receives product from the coders and holds it while the coding is being finished. So that allows us to ensure that that coding part was good. It also allows us to get all of the results back from that Bruker tandem before we kind of release that product into the good product. So that arm sort of separates out it also takes samples and separates out your sort of good and bad product in the system. So I'm going to move a little more into the control strategy for that first product. So this is an overview of how the CM controls are integrated into the overall sort of plant infrastructure. So at the top there, sort of above the box, we talk about the existing plant enterprise and site manufacturing execution system. So that brings in your high-level business controls on things like batch production planning, raw material dispensing, finished good tracking. Then we move down into the box itself. So that's where we have the controls for the process. So we have the supervisory control and data acquisition for the process and PAT data. We have the CM process itself. So the controls in green are the ones that run at a high frequency, sort of as your automatic controls on the process. So things like you see flow rate and speed and temperature on the coding process, for example. Then as we move on in red, those are the controls for where we would have potential diversion material. So there's a metal checker, as you would have in sort of any batch process, but there's also rejection from the RTD model if anything is deemed to be potentially out of specification. Similarly, at the distribution arm, you could have material diverted at that point if any of your Bruker tandem results suggest that something is bad. And then finally, the blue are your sort of in-process PAT controls, so both the NIR spectroscopy and the RTD model. So I mentioned sort of the uniqueness of our process. I'm certainly not going to go through this one by one, but one of the questions you have early on in a process is, do you need that on that NIR spectroscopy, or is something a little more simple appropriate? And this is the analysis that we did for our product, and we determined that the RTD model had advantages over NIR in this case. You could use both, right? And so that's the question, why not use both? But then you have to compare the two. What do you do when they disagree with each other? And what does comparison even mean? Because NIR is taking a small sample of the blood that it's seeing at any given moment in time. The RTD model is calculating what the average composition would be based on all of the feeder data. So they're not the same thing, and so how you compare them becomes a challenge. So with our approach, we went with the RTD model as the primary control. It has a lot of advantages, as we said, and I could probably go into this in more detail, but it's probably more detail than we need to be for this conversation. So in the opening talk, Adam mentioned RTD models and mentioned how they're in the textbooks that were written before I was born. So it's not a new concept. Any chemical engineer learns it, and so it really isn't a scary thing, even if it has a couple of scary equations. What an RTD model does, for those who don't know, is it uses the composition of the incoming materials, or I should say the flow rates of the incoming materials, so in this case your feeder data, and then using very fundamental equations, it shows you the distribution of that materials that comes out of a blender. And it's worth pointing out, so the two blenders in the system are both blenders. The tablet press also is essentially a blender. So we model the system with 3 blending units. So blender 1, blender 2, and the tablet feed frame as a third blender. And then for modeling reasons, I guess to explain simply, any real blender can be modeled as a combination of what's called a plug flow reactor and a continuous stir tank reactor. So that's a very, again, any chemical engineer who learns this, it's not some complicated concept. So moving on to the results of how we validate. So what we did during our model validation, we used 3 different flow rates that represented the design space for what we were looking at for the product. So 15 kilos an hour up to 90 kilos an hour. For each of those, we did 3 step changes in the flow rate and looked at the what we call the T10, T50, and T90. The time that 10%, 50%, and 90% of that disturbance should make it through the system. Sampled it at those points in duplicate, and then we compared results taken at all of those points by HPLC to what we got from the model prediction. The root mean square error prediction there is 1.4%, and that shows that the model does very accurately predict the concentration that we're getting in the range that we studied during the study. So then the question becomes, okay, well, you have that model prediction. How do you translate that into a control? What do you use to reject the material? The approach we took was we started with the assumption that we want 95% confidence that all of our individual tablet assays will be between 85 and 115. So, everyone knows USP criteria for releasing batches and things like that. It takes a little bit of a leap to go from what you do to translate that into the control, because they're not perfect. But that was a sort of starting point that we took with, and regulators seem to agree with that approach. So starting with that assumption, we then calculated our rejection limits, the lower and upper limits, starting with that 85 to 115, coming in by a statistical factor. And then what we used was the root mean square error prediction for the model as the power. Combined that with weight variability, because any individual tablet is going to have some variability, and that in turn gave us the assurance that each individual tablet would, with a 95% confidence, stay in that range. And it is important to note, when you think about that 95% confidence, model doesn't even matter unless there's been a disturbance, right? So most of your batch is always going to be running right around 90%. So this control is really only relevant in the time after a disturbance in feeders first restarts. So overall across your batch, you're actually much total control even than 95%. So then moving along to how we have different levels of control to assure product quality. So first you start out, you have the equipment operating itself. You've designed that equipment to produce good product quality. You've done your development. The blending is good. So you already know that that works. Then you have your process controls. The RT model is the primary control in our case. We do also have the every 15 minutes for the tablet assays. That will tell you that something really happened. It's hard to really imagine what the drift will be, but it's an assurance. You also have other things, your sort of typical controls like your weight, thickness, and hardness in the process. We also had statistical process control that we used on all of the burger tandem results, including the tablet NIR. So that would, again, show you any signs of potential drift that you might want to worry about. And then finally, you have your in-process release testing, so that includes the So here are our results from some of our what we call normal operating conditions batches. So sort of similar to what a validation would be for QBQ. And so we did that at 21 kilo an hour and 75 kilo an hour across our different tablet images. And you can see the number of points there, the duration of the runs that we did, and you can see how tight the controls are on the different variables. So there's weight, hardness, assay shown there. Moving ahead, so this is just sort of zooming in on one of the batches to make it easier to see. So here we have tablet concentration by NIR, and you can see how tight the control that was across the batches. So moving on a little bit to our regulatory interactions that we sort of took along the way to get to this point. There were several sort of progressive elements in what we were doing. I'll kind of briefly go through them one by one. So the first was there was a small formulation change in the process. So this wasn't something we 100% needed in this product, but we knew we might for other products. So we have, if you're going to have a simultaneous batch and a continuous product at the same time, you might need different grades of excipient, you might need different quantities of excipient. In this case, the formulation change was just lowering the amount of film coat on one of the images from the batch process. So it was required for operational efficiency, but it wasn't absolutely required. The next was the novel definition of the batch that we took. So we wanted to define the batch by the quantity, the quantity of acceptable liquid material produced. And so that's slightly different than what some others have taken, but the reason was if you're designing your system to be flexible to work to patient demand, you don't want to start out with 100 kilos and then only get 5 kilos, 100 kilos. So by adding in some controls that would ensure that we don't reject too much material or run in a state of control, we were able to have a system that would allow us to continue manufacturing to get our desired quantity as long as we understood the reason for any diversions and they were within sort of normal experience. We proposed a performance-based validation criteria for the NIR model. We already talked about the RTD model and where that was. I guess the other point is the difference between rejection and diversion of material. So if we rejected material from the tablet press, that material was waste. So you would combine things that were rejected by the metal checker, for example, with anything that the RTD model calculated was potentially outside of specifications. But if you had a payload of material that you wanted to divert because you had some questionable result on the NIR or the tablet hardness, that material was diverted and held, and we could, with investigation, show that material was good. We spoke to many regulatory agencies during this process, so definitely thank you to the ATP for engaging with us a lot. Here 4 or 5 times meeting with them and got some very useful information. But also the EMA as a whole, as well as the MB, the MHRA we spoke to many times. We also spoke to PMDA and Alpha. So did kind of work with a lot of the agencies about what we're trying to do to make sure that we understood that all along the way. I mentioned the divestiture of our equipment. So unfortunately, we didn't go all the way to producing this commercially because of that sort of business reason. But we did have the FDA review our filing through the form of the PACMP, and that was approved by them. So I'll slide past the slide. So what do we need to be successful in further developing PCM? I think the formulation flexibility is something where, particularly in some of these smaller markets that aren't as familiar with CM, or even, not even small markets, like even Japan. Japan has a law that says you can't do this right now.
So over time, that might be needed to switch between batch and CM. Parallel production processes. So we want that flexibility, but things become complicated, right? There is existing equipment, as others have mentioned. So if you need to make a product for some small, local manufacturer, you might be able to need to make that product in batch for that market, even if you're making it by CM for the rest of the world. Or maybe you want to make it CM there too. So you want that flexibility to be able to do what's right for patients, to respond to things like pandemics. Consistent control strategies is important. So, as I mentioned, the FDA agreed with our approach here, but we don't know for sure if every other country would approve it. And if you don't have one control strategy approved by everyone, then you have the need to make two different versions of the product for different markets, and that certainly is not giving you the flexibility. And then finally, flexible batch sizes. We got very good feedback on our approach here, and it was something that was really important to us. But knowing who would be able to get that through all markets is really important. So just sort of some quotes there at the bottom. To meet patient needs is why we do this flexibility. It's not just about the companies; it's about the patients. Having that flexibility will reduce supply chain shortages. It will increase access to medicines, and so that's why that's the why. And reliance and concepts like that from different regulatory agencies will to speed up the adoptions. So in summary, we're using CM to try to enhance our supply chain flexibility and patient access. We converted an existing batch process to CM using that novel control strategy with the RT process model, and despite its successful implementation at Merck and other companies, there is a lot of potential for further adoption of CM. This is why we're here. I think further collaboration through forums like this would really help us to lead that direction. And I just want to say, a whole lot of people, all of whom don't fit on the slide, did this work, so it's not.
So thank you, Frank, for a great presentation. And so we're going to transition now. So if I could ask our speakers to come up and take seats. And while that is happening, I want to do a very quick note to anyone that's a facilitator this afternoon. After this presentation, if you could meet us out front, we've got to go over other things in a facilitated discussion.